We continue with the pre-program.
It's a great pleasure to talk now to Professor Mark West, who lives now at the University Retirement Center, which has nothing to do with the University of California, Davis, just the name is University Retirement Center, URC. Mark.
Hi, thank you.
So I understand that yesterday you got the vaccine.
Yes, here at University Retirement Community, we have 3 levels of care: skilled nursing, assisted living, and independent living. And so I'm in independent living. The skilled nursing and assisted living people got the vaccine a couple weeks ago. But finally, yesterday, all of us in independent living got the vaccine who wanted it. And there's, over, 270 people who live in independent living. And so we all got the vaccine. We were assigned a certain hourly slot by what floor we live on. So we all went down at our recommended time, and we got the shot, and it was just like getting a flu shot.
Did you feel any reaction after getting the flu shot?
I felt fine. They had us all sit in another chair for 15 minutes after we got the shot to make sure that none of us had an immediate allergic reaction. So we all sat there for another 15 minutes. They wrote on the page what time we could leave the auditorium.
So the Yolo County people came together and they worked together with the retirement center professionals, right?
The Yolo County people brought the vaccine to the retirement center, and then our nurses who work in our skilled nursing unit were the ones giving us the shots.
Did you get the Moderna or the Pfizer vaccine?
We got the Moderna vaccine.
So that means that within 21 days, we're going to get the second.
Right, we'll get our second shot in 3 weeks. We haven't been told exactly which day yet, but we'll get our second shot.
But even after that, you are going to keep doing whatever you're doing in terms of social distance and the masks and everything, right?
University Retirement Community has done a great job. There's only 3 or 4 residents who've tested positive at all since March, in the last 10 months. And we all have to wear our masks every time we leave our apartments and go out in the hall, and we have to social distance. There can only be 3 people in the mail room at one time. And of course, they closed our dining room last March. They opened it up for a couple weeks in June and a couple weeks in September, but it's been closed. So they deliver our food to our apartments every day at 5 o'clock, because we get 1 meal a day as part of our monthly fee here at University Retirement Community. So they bring us so much food, I don't have to grocery shop at all. I save the soup and salad for lunch the next day and eat the dessert with my coffee in the morning. So we get lots of food. For the most part, it's good food. But we're all very happy to get the vaccine. And I found out today that 96% of us in independent living chose to get the vaccine. So that's really great.
I understand that the Healthy Days program is reach out to the retirement center so that you people can have also the saliva test every quarter,
we could go get the saliva test.
Have you done that already?
No, I haven't done that because, I've been here. I mean, I go out to grocery shop myself, and I go down once or twice a month to see my kids in Oakland. But other than that, I don't see hardly anybody. So I've never felt called upon to get the test, even though now there's advertisements on the TV telling us to get the test twice a week in Davis. Have you gotten tested, Walter?
Yeah, since I'm in work, we have to test every 7 days. Normally I do a Monday afternoon because it's a convenient time. And it's very easy. It takes about 2 minutes to 3 minutes at most to collect the sample.
I have a friend who lives outside URC, and she goes every week to get tested now.
All right. Okay, so very good to talk to you, and thank you for sharing your experience. We are all looking for this opportunity this time that we are going to get our vaccine.
Right. We were told we would get it by the end of January, but then all of a sudden we got a memo we were going to get it in 2 days. So we are all very happy that we got it.
Let me give you a little bit background about the quantitative polymerase chain reaction, qPCR, that's used for testing SARS-CoV-2, the virus that causes the COVID-19. It's also called the nucleic acid test as opposed to the serological test that looking for antibodies. The structure of the virus is here. The most notorious protein is the spike protein, but there are other structural proteins such as the E protein, the M protein, and also the nucleocapsid protein N that's encapsulated the RNA in the center of the virus envelope and protect this very unstable RNA molecule. The first step is to collect the sample, and there are various ways. We are going to be discussing the most comfortable way, the saliva test. And once the sample comes into the lab, first order of the day is to inactivate the virus to protect the people work with the samples. So the virus is inactivated, then the RNA is extracted. Some cases RNA extract is bypassed, and then it goes to the next step of reverse transcription. That's to say, to transform this unstable molecule into the complementary DNA, the cDNA, that's going to be used as the template for the qPCR reaction. The qPCR reaction utilizes enzymes, nucleotides, buffer, primers in the probe, and all these things can come together in one mix that's called the master mix. And the first step then is to utilize the reverse transcriptase, an enzyme, and primers to reverse transcribe the RNA into cDNA in a process initially to generate a more stable template. So here's an original template of the RNA and the reverse transcribed cDNA. Now with cDNA, we have the template. We're going to start the PCR reaction. The first step in the PCR reaction is to separate the strands of his DNA because they form hydrogen bonds. And then separate that, and the next step is to undergo hybridization. That is to anneal the primer with the sequence that you wanted to amplify. And then finally, the DNA synthesis utilizes another enzyme that's going to undergo amplification and extension. So this all occurs in one cycle. These all steps change the temperature back and forth at different times, and the reaction keeps going and repeats itself. For the primers, we utilize primers for certain part of the sequence. In the case of the United States, most of the primers being utilized are the two sets of primers targeting the region of the genome of the virus that encodes for the N protein. Other countries utilize other parts of the sequence of the virus, and for example, in the United Kingdom, one of the primers that they use three sets of primers. One of the primers for the spike protein. That's how they end up discovering that there was a very important variant in this coronavirus that is called now the British variant B117. I guess that comes from the United Kingdom, should be 007. That's why they discovered, by utilizing this the process of the PCR reaction, that one of the set of primers was not working, but the other two were working. So they were able to discover this very important variant. The primers in the United States, again, we use two sets of primers recommended by CDC. One is set N1. There's always a set of primers. There is a reverse and forward prime. The yellow represents the forward prime here, and the blue represents this reverse prime. There's two sets of primers in the region of the N protein. Now, with the template, with the cDNA that was prepared, and this two set of primers, the enzymes and the other reagents and the nucleotides, the reaction is going to take place, and every time we duplicate the number of copies that we have by one cycle of this reaction. So the more accurate it goes, the more samples we get, and initially, depends on the amount of sample that we have at the beginning, the amplification is going to go very fast, because it's an exponential process. The probes are oligonucleotides, the same as the primers, except that they have one fluorophore into one end and a quencher on the other end. Fluorophore is a chemical substance that emits fluorescence light. When we excite with certain light, it emits a light of a different frequency. And then we can detect whether a DNA is there or not by this fluorophore. How we do this is that these probes, they do not emit fluorescence because of the quencher. However, when the quencher is separated from the fluorophore, then the fluorophore can fluoresce. And how this thing happens is that this probe is going to anneal to a certain sequence of the target that you wanted to amplify. And once the enzyme is amplified, the enzymes get rid of everything that they encounter in front of it, including the probe. So once the enzyme destroys the probe and amplify that cDNA, now the fluorophore is separated from the quencher, and therefore it can emit fluorescence. And the more molecules of this fluorescence, it means more molecule of cDNA. Here's a graph that shows the fluorescence intensity versus the number of cycles, and here we see that at some point the fluorescence increases very dramatically and then reach a certain plateau. Here's another curve. Let's see that this was two separate patients looking for one of the two primers, set of primers here like N1 or N2. On the left, you see that the numbers of cycles was much smaller to get to a certain threshold than the sample, the blue sample on the right side. So it means that you started with a larger number of copies. For example, if you get a patient that has a larger number of copies, a high infection rate, that patient might produce a CT that's very low compared to another one that's still infected but not larger number of copies. CDC determined that the threshold for that is 40 cycles for N1, N2, and also for a control that they use. The control that they use is a positive control, is a human RNA-ACP. RNA-ACP is a RNA, is also an enzyme, and it's very common in any part of the body. You can collect that enzyme. So if you collect the sample and we collect this RNA-ACP, then we are going to be able to check the quality of the sample. And also they use a positive control. The positive control is more to see the quality of the work of the reagent, the lab itself, if everything is functional. We use a non-infection form of this SARS-CoV-2, and that's only to determine if there is a level of detection that's appropriate in the lab setting. The interpretation of the results is such that the threshold value is 40 cycles. So if both sets of primers for N1 and N2 gave smaller than 40 cycles, regardless of, we don't even to have to look at the RNA-ACP, you can conclude that this is a positive. This came from a patient with the coronavirus, because the number of cycles was not, didn't need many cycles to be able to come to a certain level of fluorescence there. Now, the opposite of that is a situation where you need so many cycles, maybe more than 40 to reach the threshold. That means that this is not significant. This could be a background signal from fluorophore itself. However, you have to look at that point what happens with the CT from the RNA-ACP, because it tells about the quality of the sample. If you have a CT less than 40 for RNA-ACP, that means you have a good quality sample, but we could not find the SARS-CoV-2 virus. So therefore, this result is negative. Now what happens is if both curves are above 40 and the CT of the RNA-ACP also is above 40, it means that this is not a good quality sample. So you cannot conclude whether this lack of evidence for the SARS-CoV-2 virus is because the sample is not good or is because it's not there. So this is inconclusive, this is invalid results. You have to repeat these results. The same thing if you get a CT above 40 and for one of the set of primers and for the other set of primers a CT below 40. This is inconclusive, it's inconsistent. Both set of primers should amplify equally, so it should not be considered a sample. So this is a very simple summary what we do. There's much more detail to that, but but I hope that this background information would help you to understand the more complex, high throughput system that we are going to be talking about today.
The official program starts at 5 o'clock. We are running some pre-program. As you know, this format here includes questions and answers. And the questions may come in two different forms. The questions that have been received by electronically by the registration time in the registration form and other ways. And some of these questions, I ask people to record that question in a video. So we're going to have video questions, and we're going to have a regular question that I'm going to display on the screen, and the latest minute questions that there was no time to prepare the slides for that I'm going to read here, such as questions coming from the Q&A. One example of these questions that are going to be video questions, one participant from Nairobi, Kenya, asked a question, and I asked him to prepare a video of that question. Here it is as an example.
My question is that the saliva tests, just like the nasopharyngeal test, are both invasive. And I was just wondering whether anyone is thought about perhaps looking at less invasive techniques such as breath odors, because that captures both odors from the mouth as well as the nose, and then identify potential biomarkers, and then the use of less reagents directly with mass spectrometry, analysis, gas chromatography, mass spectrometry. Any thoughts on that? Thank you.
So for this kind of questions, different questions, there are different specialists on campus. So when I got the questions, I looked for specialists on campus that will be able to respond to these questions. And for this particular one, we have a specialist here in this area, Professor Oliver Finn. And I asked Professor Oliver Finn his thoughts about this question.
So in general, breath analysis is used. As we all know, for driving purposes, you should not drink alcohol, because the ethanol itself is a clear biomarker that would tell an officer, a police officer on the street, whether or not you had recently drinks that contained alcohol. Similarly, other volatile compounds are being tested regularly by ion mobility spectrometry. For example, at every airport, when you go through these machines that blow gas at you, then it has been tested for specific markers for example, explosives. So these are two practical tests where volatile compounds are being routinely tested with very high efficiency, very high selectivity, a very low false positive rate, and also very low false negative rates. The question now is, can we do similar things for breath analysis for COVID-19? Here we do not know any specific organic compound that is volatile that would be produced after this kind of viral infection that would be different from other viral infections, and that would be so specific that an instrument, be it a mass spectrometer or an ion mobility spectrometer, could differentiate it from local noise. So we do not have current biomarkers. We do not know what we're looking for. And clearly the viral load is low compared to other chemicals that we have in our mouth or in our lung system, respiratory system. For example, foods that we have eaten, spicy food, people who smoke, they have abundant volatile compounds that are easy to be detected. So any kind of volatile from COVID-19 would be more specific and different to all those thousands of compounds that we have in our mouth and our nose and our respiratory system. So that is not only looking for a needle in a haystack, it's actually not even knowing how big the needle is and what kind of material the needle is, what form or shape a needle would be. And I think currently that's pretty much hopeless.
So there were many questions that are very similar. So if you don't see your question there, probably you are going to see a very similar question. So when I find these questions, either I put them together as a group or I use one as a representative question. I always give you priorities at the question on the way that they arrive, first come, first serve base. And I'm going to display those questions during the presentation. We have a very good program today. We're going to be discussing about tests and the different types of tests. And we're going to be talking about vaccine. And we are very blessed to have the opportunity to have Dr. Stuart Cohen, because he's in the front line. He's doing clinical trials, not the only clinical trials with vaccine, which is important, but also clinical trials with treatments. And he has so many important information to share with us today. He's a very, very busy man. So that was, I'm very glad that he was able to accept in a short notes, because initially this was planned to be a symposium on testing. And when the questions arrive, we are not going to ignore the question that people are asking. So we started seeing too many questions about vaccine. So we updated the program and we asked Dr. Stu Cohen whether he would be able to attend. And he made an effort, and he's going to be able to be with us today. So that's going to be a very interesting program. I urge you to stay up to the end. There will be many interesting questions and answers and the different topics to be discussed. Like I said, questions in format of video questions, some other questions in the format of a display on the screen, and I'll read these questions and they ask one of the panelists to respond to them. So this is going to be a very interesting program. It's going to take a little bit of your time, but I hope you are going to the end of the day, you are going to learn a lot and be well informed. This is a very important pandemic going on now, and the information and the correct information is very important. So we are going to start at 5 o'clock. We still have a little bit of time, and we end here the pre-program phase of the symposium. Start at 5 o'clock. We finished the pre-program and we're going to start the program at 5 o'clock. Stay tuned. We start in 1 minute. We just finished the pre-program and we're going to start in 1 minute at 5 o'clock. Start in 30 seconds. Good afternoon. My name is Walter Leal, and I'm distinguished professor in the Department of Molecular and Cellular Biology here at the University of California, Davis. I'm going to be your moderator today. I would like to welcome all of you that are joining now or maybe join later via Zoom, YouTube, or Twitter. We have a very good program today and the participation of 1,200 participants from 13 different countries, including the United States. I hope you enjoy the program today, which will include Professor Richard Mitchellmoore, Dr. Nantra, Professor Heather Bishoff, the Mayor of the City of Davis, Gloria Partida, Dr. Oliver Finn, who spoke in the pre-program, and Dr. Stuart Cohen. Without much further ado, there are also, I wanted to mention that there are 2 surprises for you at the program, and I hope that you stay tuned towards the end of the program. Without much further ado, I'm going to invite 2 undergraduate students that are going to introduce the Chancellor Gary May. The students are Kelly Brandt and Banin Alof.
My name is Kelly Brandt.
And my name is Benina Lofi.
We are undergraduate students at UC Davis, currently enrolled in Dr. Leo's biochemistry class, Biz 102.
We are honored to have the opportunity to introduce Chancellor Gary May.
Dr. May became the 7th Chancellor of the University of California, Davis on August 1st, 2017. Dr. May came to UC Davis after a 3-decade career at the Georgia Institute of Technology in Atlanta. He was the Dean of the Institute's College of Engineering, the largest and most diverse school of its kind in the nation.
Here at UC Davis, Chancellor May leads the most comprehensive campus in the University of California system, with 4 colleges and 6 professional schools. UC Davis enrolls more than 39,000 students, brings in nearly $850 million annually in sponsored research, and contributes at least $8 billion annually to California's economy.
Chancellor May is a native of St. Louis, Missouri. He received the BEE degree in electrical engineering from the Georgia Institute of Technology in 1985 and the MS and PhD degrees in electrical engineering and computer science from the University of California, Berkeley in 1987 and 1991, respectively. In 2010, Dr. May was named Outstanding Engineering Alumnus of UC Berkeley.
At the Georgia Institute of Technology, Dr. May developed nationally recognized programs to attract, mentor, and retain underrepresented groups in the STEM fields of science, technology, engineering, and math. For his unwavering commitment to undergraduate education, President Barack Obama bestowed on him the Presidential Award for Excellence in STEM Mentoring in 2015.
Dr. May is an accomplished scholar and engineer. In 2018, he was inducted into the National Academy of Engineering for his innovations in educational programs for underrepresented groups in engineering and his contributions to semiconductor manufacturing research.
Last year, Dr. May was elected to the American Academy of Arts and Sciences under the classifications of educational and academic leadership.
Please join us in welcoming the 7th Chancellor of UC Davis, Dr. Gary May.
Dr. May, the floor is yours.
Thank you, Walter, and thank you, Kelly and Benin. That's a really wonderful introduction. On behalf of the university community, I'd like to welcome everyone to our fourth UC Davis COVID Symposium. This series was launched in April, just weeks after the COVID-19 pandemic took hold here in the United States. And since then, thousands of viewers from around the world have heard from UC Davis experts, civic leaders, and public health officers. Their information about COVID-19 is vital to the public as we endure these challenging times and new questions seem to arise every day. Previous symposia have focused on such issues as vaccines and how our health system is managing the pandemic. Today, we'll focus on an especially critical aspect of the COVID-19 fight, and that's testing. Through widespread testing, we have a better chance of stopping the spread of the virus, and it can also help those who test positive to get care or isolate sooner. Today's symposium is happening as we have reached a testing milestone. On January 3rd, the UC Davis Genome Center had processed 100,000 saliva samples in its testing of asymptomatic people over the past 3 months. What a remarkable achievement. This rapid saliva-based testing program launched in September with students and university employees. It has since been expanded to Davis residents through the Healthy Davis Together program. This is a joint initiative between UC Davis and the City of Davis to prevent the spread of COVID, and it's been a great success. The community benefit of having a major research university with a genome center couldn't be more apparent. Still, many of us have questions. How reliable are saliva-based tests? How are the results able to be processed so quickly? Could saliva-based testing replace the nasal swab for good? A panel of UC Davis experts is ready to answer these questions tonight and share more important information about COVID-19 testing. They'll also detail other measures we've undertaken at UC Davis in the fight against COVID-19, such as hospital and wastewater surveillance testing. We may also have the opportunity to branch into other topics as well, including the latest information about vaccines. I know that's of great interest to many of us. I appreciate everyone's participation today and wish you a happy new year. With the advancement in testing and the availability of vaccines, I'm optimistic that 2021 will be a much better year for us all. Please enjoy the symposium.
Thank you very much, Johnson Lamey. We will continue now, and let me get back here to the presentation. So the format of the presentation is going to be that we are going to have questions that were being asked either via a slide display as this one here, as well as video questions. For example, this question here from Dana Armstrong. She asked the question, Dana asked, can you explain what a PCR test is and how it's different from a PCR test or antigen test? So there's a confusion there about qPCR, PCR antigen test. Another question from Summer Kalvach. If you are no longer infected, there will still be antibodies present to prevent a future infection. So won't the test come up as positive for infection, even though you are not? So these are very common questions that came in many times. Those are representatives of multiple questions that we receive in the same line. So let me explain very briefly to you today and give a background for you to understand the more complex things that are going to be discussed today. The virus is RNA virus. When you get infected, the virus will replicate in your body, would shed, would be put out. You'd make more copies of RNA and the proteins that are necessary for the virus. So during the process of infection, there is a boost in the amount of the RNA and the antigens, the proteins from the virus. And your body starts making antibodies that Dr. Stu Cohen is going to talk about later today. And these antibodies first start with the IgM, and then IgG kicks in. So during the process of infection, let's say around 10 days, there are no antibodies there to detect it. And after the process of infection, there are no RNA to detect. So for the test that you want to do, depend on what you are looking for. If you are looking for antibodies to using for a program to use this antibody to rescue other patients, then you are going to be necessary to use the antibody test. But if you want to look for what we are most interested in, that's patients that are asymptomatic or patients that are having COVID-19, So that we wanted to look at the nucleic acid test, the qPCR test, because the virus is replicating, is shedding, so there is enormous amount of RNA available. And for the other convalescent plasma and other programs, we want to know if the blood of that person includes antibodies that may be used to rescue other patients. So these are two different types of tests, and they look for two different things. So answer the question from Summer, if you have an antibody, you are not going to be interfere with the results of the RNA that you are looking at in a qPCR program. So the virus, like as explained many times, and Dr. Cohen talks about that later on today, is an RNA virus. So the RNA is protected inside of this envelope, very well protected there, because RNA is a very unstable molecule. So when we want to detect whether you are shedding this virus, first thing that you have to do is transform the RNA in cDNA. It's reverse transcription, we call it. So you use a special enzyme as reverse transcriptase to transform the RNA into cDNA. Your body cannot do cDNA or cannot transform this virus. So these questions about where the virus is going to be incorporated in your genomes make no sense in molecular biology. So once you do that, now you have the cDNA as a template, and you can amplify because the number of copies is not big enough that you could detect an infection. So that's what we call the polymerase chain reaction. There are three names that are going to be listened all the time when we talk about these tests: RT-PCR, qPCR, and RT-qPCR, which is the most accurate of the three terms. Here's the sequence of the genome of the virus. It's short of 30,000 bases. All we needed to detect the virus to detect one part of the sequence of the genome. In this case of the United States, we use the part of the genome that encodes for the N protein. So we design primers and we look for that sequence so that you can amplify that sequence. In the United Kingdom and other places, they are using also primers for the sequence that include the spike. That's how they were able to detect earlier on the N501Y, that a variant that everyone is talking about is the variant called B117. Also, the United Kingdom did, in addition to tests, they did also sequence of the positive case in a much larger scale than we are doing in the United States. So the PCR reaction itself is a three-step of reaction that requires different temperatures, enzymes, and things like that. And they that's why we call it the thermal cycling, because it changes the temperature in three steps, and that's what takes time to do that. First, the cDNA has to be separated because they form what we call the hydrogen bonds. You have to separate these molecules. Then we have to anneal a temperature appropriate for your primer, that is a piece of the sequence, to anneal to get to your template. And finally, the enzyme is going to do amplification and extension of your sequence. So these three cycles are going on, and you use also probes, fluorescence probes, to detect those the cycles. And we need a number of cycles to go above the background of the fluorescence that already exists there in the reaction. So that's called the threshold. That's the number that we are going to hear a lot today and throughout this pandemics, is the CT values. I'm showing here two different curves for two different samples. One sample on the left side in red color that have more RNA of the virus in the beginning, and another one on the right side, the blue sample that had less RNA to get started with. Both of them amplified and they come into a threshold cycle. In the case of the United States, we are using the cycle threshold of 40 recommended by the CDC, and anything that's getting is smaller than that. The smaller the CT, that's something that you have to keep in mind. The smaller the CT, that means the higher level of infection. So when you hear someone talk about the CT value, that's what it's all about. So I think this would be a very important background information so that you can understand the more in-depth discussion that we are going to go on now with Professor Richard Mitchellmoore. And for that, I would invite another undergraduate student, Nina Jorgensen, to introduce Richard to the audience.
Hi, my name is Nina Jorgensen. I'm a student in Dr. Leo's Biz 102 class, and today I have the pleasure of introducing Professor Richard W. Mitchellmoore. Full disclosure, I am an undergrad in his lab, which is full of super cool people and super cool research. So after studying natural sciences at Cambridge, Dr. Richard Mitchellmoore joined UC Davis as a faculty member in 1982. Dr. Mitchellmoore was the founding director of the Genome Center at UC Davis in 2003. Since then, he's overseen the recruitment of over 16 genomics and bioinformatics faculty, along with the development of 5 technology service corps that enable genomics, proteomics, and metabolomics research on and off campus. He's currently a distinguished professor in the departments of plant sciences, molecular and cellular biology, and medical microbiology and immunology. In fact, Dr. Mitchellmoore has already published over 200 scientific papers. His interests span basic research into the molecular basis of specificity in plant pathogen interactions to translational plant genetics and crop improvement. His research is focused on comparative and functional genomics with an emphasis on plant disease resistance and pathogen variability. In addition, his program coordinates and hosts the bioinformatic component of the Composite Genome Project. Dr. Mitchellmoore is interested in taking next-generation DNA sequencing approaches to all areas of biology. He aims to exploit such approaches for information-driven deployment of resistance genes in crop plants, which would help provide more durable disease resistance. As it may go, he's interested in fostering research that enhances global food security. Please join me in welcoming Dr. Richard Mitchellmoore, who's leading saliva testing at UC Davis.
Thank you, Nina. Richard, the floor is yours.
Thank you very much, Nina. Wonderful introduction. Let me then share my screen. Here we go. Why is it there? Right. So thank you very much for all joining us in this talk. I really want to give us a very brief thumbnail sketch of the testing effort at Davis, but this has really been a campus-wide effort with input from many people. But our goal was to provide rapid, inexpensive, high-throughput testing for the SARS-CoV-2 virus using technology that I was familiar with from the ag biotech sector. So the talk's really split up into 3. One is to give a background and a high-level overview of what we're doing. Then we can have a Q&A session. I've got validation results from the Davis test, and also going forward of where we are going to take the testing, particularly as vaccinations become more common, and what are we going to do about the new strains. So as Walter has just described, we run a nucleic acid test. This is not an antigen test that Dr. Nam Tran will probably talk a little bit more about later. It's a real-time reverse transcriptase PCR. And we have RNA in saliva, we don't extract it, and then the reverse transcriptase makes DNA, and then we have the PCR reaction. You don't really need to know the details of this, except that every cycle of the PCR, a fluorescent probe is activated. So this allows us to monitor the amount of DNA that's made in each cycle. And the question here is, are the results clear? People have asked me this. Well, here's an actual trace. We run samples in batches of 760. And here we look at 2 viral genes. One is shown, M1 is shown in green, the other one in purple here. And you can see the negatives. They flatline. There's no signal there at all. And then pairs of positives are coming up. So the answer, yes, the readout is pretty clear. And as Walter just said, that you get a different CT value, you get different indicative of different amounts of virus. And here, with a low CT value, you've got lots of virus. Here, right at the top here, after about 32, 33 cycles, you've got much, much less virus. And then if you haven't got any virus, it flatlines. So in answer to people's questions, is it clear? It's clear. So this is a really important slide I want you to understand about what testing means. You need to think about what happens during the progression of the disease, whether you're, or the pathogen cycle, whether you're symptomatic or asymptomatic. And if you're dealing with community grade surveillance, that is testing of asymptomatic individuals, that's slightly different than clinical grade testing. We're thinking about detecting infectious individuals rather than detecting everybody that's potentially infected. What we need is a rapid, inexpensive, high throughput workflow. Sensitivity to some extent is secondary to frequency and turnaround of results. If we have a very, very sensitive test or slow turnover results, it's really a waste of money and resources. And the reason for this is that after you're infected, it takes a couple of days before you get any detectable virus. And then it very rapidly climbs up to large amounts of virus and then goes down. And your most infectious are about, I don't know, six, 7 days at the most. But you then have this long tail where you have nucleic acid, it's detectable, but you're no longer infectious. Now, also in terms of being infectious, you know, very conservative threshold, a CT value of 32. Some people will argue it's more like 29 or 30. The FDA calls a positive call threshold of 39. But you know, we have a test that really is, our limit of detection is similar to the conservative threshold for infectivity. Now, if you detect an infected individual at the peak here, you send it off to a commercial company and they take 7 days to return results, and the contact tracing goes back 2 days, it's a waste of time. So slow return of results, the only people who are really benefiting from that are the testing companies. So I was aware of technology in the agricultural biotech sector. The companies regularly will screen hundreds of thousands, if not millions, of data points a day. And the way that they do that, particularly the Nexar in the middle here, is it arrays PCR reactions on tape. And then these are set up and run. There's a smaller system that we actually went with, the IntelliCube. Now that is a qPCR platform. It sets up the reactions and runs them and gives it the results. So we, in collaboration with LGC and Corteva, we've now got 2 IntelliCubes, and we adapted the FDA approved screen for N1 and N2, the 2 viral genes. And from that, we've been able to put in place a very rapid, inexpensive, high throughput system. And this was rolled out in October, and it was really high throughput testing. There was parallel nasal swab testing for short term while we calibrated. And now we have quarantine and contact tracing. Initially, we did 1,000 students. It ramped up to 2,500 the second week, 4,500 the third week. And now we've expanded to all students, faculty, and staff with at least weekly testing, with focused testing for potential outbreaks. We then went from 21,000, and now we're looking at 50,000 tests per week. And as you'll hear too, we've expanded to the city of Davis through the Healthy Davis Together program. This is what our testing, our sampling kiosk looks like. This was a tremendous effort. It's now taken over 4 basketball courts. There are 38 spitting stations. We sample 7 days a week, 8 hours a day. The capacity is for like 32,000 samples a week if there was running flat out. And the time in and out is really efficient. It takes people about 4 minutes. We've replicated this concept of the kiosk with a Mondavi theater for Healthy Davis Together. And they have some smaller operations as well. So the way it works, you make an appointment online, you receive a QR code, you show up at the kiosk, then the QR code is matched to a barcoded tube, and then you take that to the spitting station and you're given a small amount of water, you rinse it around your mouth, you spit in the tube, and you leave. There is absolutely no PHI, no personal health information that moves from the kiosk into the testing lab. So we then do run the test, and then we report the result back for that tube. And then in the HIPAA compliant environment, the result is matched back to the individual. We only screen asymptomatics. The symptomatics are sampled elsewhere with a nasal swab. This is the lab, the two IntelliCubes. It's a spin-up lab. It's a CLIA certified extension of the student health lab. The workflow looks something like this. People have asked, what happens to my saliva? Here's the tube. We accession them in 96 tube racks. There's a barcode on the bottom that is read automatically. It is heat inactivated. The virus is readily heat inactivated, which is good. We have a robot that decaps 96 at a time. It's a great machine, but it doesn't always work, but it works most of the time. Then we aliquot protease. One of the big challenges of working with saliva that a lot of people were wary of was it's very viscous, or at least some people's saliva is. We solved that problem by using papain, or AKA meat tenderizer. If you eat papaya, your lips tingle. That's because papain is doing a number on your saliva. And when we add a small amount of it to the tube, it completely solves the saliva viscosity problem. And then we can aliquot it from the tubes into 384 well plates. We put 2 of these in here, and then this machine, the IntelliCube, will automatically set up the reaction and run the PCR. And then from that, we get the results. Actually, there's a program that we export the results to the cloud, and it then uses artificial intelligence very sensitively to call the positives and negatives. So that's the end of my preliminary talk. I would make one shout out though. This has been a massive team effort from all across campus. It's not just been one individual.
Thank you, Richard.
Q&A, first session.
Okay. Thank you, Richard. Can you stop the present so that I can continue from here? Thank you very much. And let me go now to segment 2 of our event. And so one of the questions that we got is how does the rapid bedside COVID versus flu test work? So for that, I invited the Dr. Nan Tran. He's an associate clinical professor and director of clinical chemistry, spatial chemistry, toxicology, and many other many other functions in his portfolio. He's at the UC Davis Medical Center, and the Dr. Tran is a product of UC Davis students. Here's alumnus from UC Davis. So I asked him to start by explaining his experience here as an Aggie.
Yeah, absolutely. So I'm very proud to be an Aggie, right? And so I was a transferred to UC Davis in 2000, fall 2000. Entered as a biotechnology major, which was a fine program, but I was very interested in molecular and biochemistry too. So I converted over to a biochem and molecular biology major. So that's what I graduated with in 2003. Have fond memories of biochemistry classes, which including the molecular side, which, believe it or not, really aligned with what we're doing today with the current state of the pandemic and so forth. So it set me up to be where I'm at today, and I really have a lot of good memories and really a lot of things I attribute to UC Davis and my success.
So who was the instructor when we took the BIS 102?
Dr. Hilt. So my understanding that he's still around, and I still remember his class with one midterm and one final. So we got to get do well on both, right? So, but I do remember it was a very good class. He's an excellent teacher, and and I now teach at UC Davis too. So I mirror a lot of the good teaching examples from Dr. Hilt and many others in my my course, which is a bioengineering course.
That's great. Now, the question that the people is asking, what's about this test for discriminating between COVID and the flu, and how it's possible to get it so quickly?
Yeah, so UC Davis Health, we deployed one of the first, actually the currently only RT-PCR platform, and PCR, a lot of people have heard about that technology. So it's what we accept as the gold standard for detecting many viruses, including influenza and SARS-CoV-2. So our platform is a point of care device, means that it can be used by a lay person, could be a nurse, could possibly be a technician, could be a physician in the emergency room, so not a laboratory technician or scientist. But in this case, the technology is able to thermocycle. So with PCR, you need to heat up, cool down very quickly. In this case, the chemicals that are used, the reagents that are used, in addition to the technology, it's able to do that very quickly and get results back in 20 minutes.
Thank you very much. I want to remind you that I have two big surprises for you today. Stay tuned. We have many questions that are in the format of a video question. Here's one for undergraduate students.
Hi, my name is Bryan Carter, and I'm an undergraduate at UC Davis, currently enrolled in Dr. Leal's biochemistry class. I heard that UC Davis is testing wastewater for COVID-19 and using that to determine outbreaks. How effective is this practice, and how can it be traced to a specific spot on campus?
That was a good question because it gives us an opportunity to invite Professor Heather Bishoff through the Health Davis Together initiative, and we're going to be talking about Health Davis Together. Dr. Bishoff co-leads the environmental monitoring program with Davis Call UC Davis Genome Center to monitor SARS-CoV-2 in wastewater, surface sample, and the HVAC system throughout the city of Davis. Heather.
Hi, Bryson. Thanks for your question. My name is Heather Bishoff. I'm an assistant professor in the Department of Civil and Environmental Engineering on campus. And yes, we are, as you heard, monitoring SARS-CoV-2 in the sewer system on campus and in the influent to our campus's wastewater treatment plant. So for those of you who aren't familiar, this is intended to serve as an early indicator for outbreaks on campus. And you asked, how effective is this technology? Well, so what we do is deploy autosamplers out into different nodes on campus. So manholes that collect wastewater from buildings. We target individual buildings with a focus on the residence halls. We can put samplers out into the manholes or into building cleanouts, and those samplers collect a portion of the wastewater flow every 15 minutes over a 24-hour period. And we take those samples back to the lab, we concentrate them, extract the genomic material, and then use PCR to detect the virus in the sample. So if we get a positive signal from one of these samples, what we can do is then go back and work with the campus to first of all, identify whether or not we know somebody has already been within a building and identified as a positive case within a building and moved into isolation or to quarantine if exposed in close contact to those individuals, in which case that explains our result. And if not, then we can send out, the campus sends out a communications email asking students within that particular building to go back and get another individual test as soon as possible to help us identify any potential positive cases that hadn't yet been detected by the individual testing system. So the samples that we collect are from buildings. They don't have any identifying information. They don't allow us to contact trace or know who is an individual that is infected, but our samples are very sensitive. So if there's one person who is sick within a building, we can detect that signal in our sample. And they're also serves as an early indicator. So often about half the people who are infected with the virus are excreting the virus in their stool. So if someone is excreting virus in their stool, they do that before symptoms are showing. And so it's a benefit to be able to monitor the presence of the virus that happens to show up earlier than symptoms do. So if you do end up getting an email from campus that asks you to go back and get testing kind of on a greater frequency than the weekly testing you're already doing, that is to help us try to eliminate any potential exposure from unidentified cases. This is a method that's being used in cities and campuses all over the country and now all over the world, but our campus is especially advanced because we have access to these individual tests that allow us to then take action from our samples and help to prevent outbreaks from happening before they occur.
Thank you, Heather. So we have more questions, and the questions from other undergraduate students as well as other professors and the public in general. This question here came from Robin Lee. Are the rapid bedside tests mostly for the emergency room? What about the regular patient in the clinic? And I asked Dr. Nantran about that.
That's correct. And so we do have a small number of these fast systems deployed at select clinics. These are for situations where a patient does actually show up, but the majority of patients that feel like they need to be tested, they actually go through our drive-through collection sites. So you drive up, you make an appointment, you drive up, you get collected, and hundreds of samples are collected each day. And we're able to test these on a laboratory system using similar technology to that 20-minute test. It takes 3 hours, however, but at the same time, we're able to run in batches of 94 samples every 3 hours. So you can imagine that we're not able to do, we traded off speed versus throughput. We've been testing quite a lot, and it's tested to the point where we're many samples, and what we found is that with the recent surge, we're testing between 700 to 800 samples during the peak. Obviously, the last couple of days with the holidays, things have slowed down a little bit because patients may not have come in for various reasons. But this is a mix of people who may be symptomatic. This is a mix of people who need testing before a procedure. So we're just making sure that they are negative before they undergo an elective surgery, for example, and many other reasons too. So the numbers have increased significantly. And we're very lucky that we have platforms that are able to handle with robotics to be able to do this automatically and handle the throughput. As always, we're always keeping an eye on how much reagents we use because, there is still a limited supply of these.
Thank you. Undergraduate students are very engaged in this symposium and they ask many interesting questions.
Hi, my name is Elise Ludigiani. I use she, her pronouns, and I am currently an undergraduate enrolled in Dr. Leal's biochemistry course. My question is, how does the false positive or negative rate of the saliva test compare to the rate in the nasal swab test? Is the saliva test purposefully more or less sensitive than the nasal swab test? Thank you.
Another question in the same line came from Sarah Watterson. I have heard conflicting answers as to whether the UC Davis saliva test is as accurate as the nasal swab. Is it? I have also heard that if you are symptomatic, the saliva test is not as accurate. Is this true? I was under the impression that the city and the UC Davis did not want symptomatic people possibly infect others at these public test sites. And that was the reason to only use the saliva test if you are asymptomatic. One more question in the same line that leads to Richard for the next segment. The question comes from Vinci. And the question is, I would like to know the sensitivity and the specificity. Many people ask the same question of the test is used for Health Davis together, particularly the sensitivity or the potential for false negatives. I would like to know how the sensitivity for this test compared to other PCR tests with different collection methods, such as the nasal swab. That leads us to the next segment for Richard to talk about the validation and the results and respond to these questions. Richard.
Thank you. Let me share my screen again. Okay, you should be seeing that. There we go. So very good questions. I'm just going to skip through a couple of slides and I hope we'll address many of those questions. And if I remember, because there are quite a few there, I'll try and address the ones that I don't. So in terms of sensitivity, you can think about sensitivity in a couple of different ways. One of which is what is the limit of detection of the test? And the way that we determine that is that we have deactivated virus and we spike it in at different concentrations into negative saliva. And the FDA requires you to do 20 replicates at each concentrations. We've done it on both of our machines. And we have a limit of detection of about 6 copies per reaction, which means it's about 15 copies per microliter of saliva at 96 sensitivity, which is what the FDA considers the limit of detection officially. That doesn't mean that we can only detect this. In fact, more than 50% of the time, we'll detect 3 copies per microliter of reaction. right? But as they define the limited detection as 96% sensitivity. Now, how does that compare to other, I'll just look at the reproducibility. The other thing you have to do to validate any test is you have to get clinical samples that have been validated or tested at other locations. And ASU, Arizona State, has saliva samples, a very similar test to the one we ran. We got their samples, so it's 47 positive, 47 negative. The FDA says you've got to have at least 30 of each. And we ran those samples, and what you can see that all the negatives were negatives, there were no false negatives, and all the positives were positives. So there were no false positives or false negatives. And then if you look at the CT value, you've got this wonderful correlation. So our test is detecting almost exactly the same amount of virus as the Arizona test did. And actually they were looking, it was a different test, they were looking at different viral genes. So it gives us a lot of confidence as to the accuracy of our test. The other thing, I should go back up here, in terms of comparing with other tests, there is actually very little data on sensitivity of saliva tests, but you can compare it to the LODs, the limits of detection of many of the EUAs issued for swab tests. And there it's comparable. It's a little bit less sensitive than some of the declared swab tests, but the swab test is very different because they're sticking something up your nose, they're running it around, and then they put it in a solution. Here we have your saliva. We know that this is the number of copies in your saliva. So it's very difficult to equate the two. But basically, people have seen a very tight correlation over multiple studies now between saliva and the swab tests. Though as the disease progresses, it may fluctuate slightly. But clearly, there is a lot of virus in saliva, and it equates pretty closely to if you're high in a swab test, you're going to be high in a saliva test. All of this data is put out for people that are not familiar with the Davis system. You can go to the website, and there's a lot more data here that I encourage you to look at. This is actually an interacting scheme, so you click in it and you can get a lot more data. In terms of the turnaround time, we're now returning about 95% of our results the next day. And as the Chancellor mentioned, we passed a landmark, but we've now got over 130,000 tests for Healthy Davis and the campus. Over 31,000 unique individuals have accessed our test, and we've detected just shy of 1,000 positives. This means that the rapid tests, we turn it around, we've taken out of the circulation nearly 1,000 infectious individuals, and we quarantined the close contacts. So getting back to the scheme again, in terms of sensitivity, yes, there are some slightly more sensitive tests on the market, but in this grand scheme of things, it's a very short window, maybe just a matter of hours, where you'll have a very sensitive test, will detect infection where our test won't. So the Davis test here is about 15 copies per microliter. We'll definitely detect anyone that's infectious. And then in this long tail off, our test will drop away faster than the most sensitive test. So why do we run a saliva test? We run a saliva test because people don't like having things stuck up their nose repeatedly. And there's plenty of virus in the saliva. It is for asymptomatic individuals. It has nothing to do with the saliva. It's just that we don't want infectious individuals showing up to the kiosk and potentially infecting people. So it's very important if you're symptomatic that you log in and you'll get tested with a nasal swab. And in many cases, it'll be in your car. You don't get out of your car. And so there isn't a risk when you go to the kiosk that you're going through the queue where there are lots of other people that are potentially infected. Back to you.
All right, thank you.
I hope I answered all the questions.
Yes, thank you. And let me start continue sharing here. Go to segment 3 now. All right, so we have some surprise to you today, but let me talk now about this question that came multiple times from different people. And I use the K1 question as an example because it was the first one in the same line. K1 asked, I am a graduate student who has been to the saliva test at ARC of UC Davis several times last quarter. I know there are many students waiting in line to get it tested sometimes, and there are also many people inside the test site. Since the test site is in a closed area of ARC, not the open space outside, I'm concerned about the chance of getting infected during the test if there happen to be someone near me who carries the virus. So because of these questions, and also because we have an audience for different countries, 12 countries participate today, and a huge number of people attending out of the state and out of the city of Davis, probably they have not seen the test. So I put the camera on and I want to take this test and show you how this thing works. First, like Richard mentioned, you have to have a survey, say that you are asymptomatic, and you have to show the gentleman here at the entrance that you have the survey that you certify that you are asymptomatic. Then another person is going to check whether your barcode that Richard mentioned, and the walking is a long walk, and you see is everything 6 feet apart. It's a huge place. It's a closed environment. Yes, it is, but everyone is walking 6 feet apart and as fast as we can, but always maintain the distance. And you see that people are very disciplined in that. And finally, before you enter the testing site, you are going to be checked again for your barcode that Richard mentioned. That's very important to have this barcode. And the people are going to be having the opportunity to watch videos explaining, especially for those who are there for the first time, what's the procedures going once they enter the ARC itself, this testing site itself. So here's in a high-speed video to show that there's confirmation. And now we are finally inside of the area that Richard showed as a photo, a still photo. Here's the site. We walk there, and there is people again checking because it's very important to have this barcode with you. No barcode, we can't do anything because that's the whole information that's going to be important to know your email and everything. So here's real time. And I'm going to show you that it takes only 2.5 minutes. The lady checked me and they asked me to hold because the station is being cleaned. I sanitize every time that one person is there. And as you see here, there is no one employees or anyone less than 6 feet apart. You come to the station, first thing to scan the barcode. And for people in Davis, they already seen this many times, but for people from outside, ask your date of birth and your name for confirmation. Make sure that everything's okay. Scanning the tube, ask you to wash your hands, sanitize again with alcohol, and get your tube.
Thank you.
Move to the next station. This station here that will be the tube, and they go there and they get a little bit of water and a pipette tube. The lady delivers to you, and they ask you to go to the next place where the saliva is going to be collected.
Thank you.
So now it was number 13, that I 14, and then I'm moving to that direction. As you see again, huge distance between people, and the protection for the employees, glass protection with fibers. And now I'm coming here to the real place where I'm going to take the real test. The collection of the saliva. So she sent me to that station. And forward to take another one because that one was now question, have you done this before? If not, you need to start to take the water, put it in your mouth, about 200 microliters of water, and to generate more saliva. Then we are going to deliver using a pipette tip, 1 milliliter pipette tip, I guess, and put that in your mouth and deliver this saliva there back to the tube. You see no direct contact with people, shorter distance. Make sure that the amount is within the required limits, not too low, not too much. You are good.
Okay.
Go, clean up, wash your hands again, and then we go out with this test is done. So 24 hours you receive that. I never received that more than 24 hours. So that's a very fast one. And I continue with the question here. Anthony Cole asking,
can I jump in, Walter?
Just 1 second, Richard. I'm going to back to you in a minute. Is UC Davis doing any outreach program to business to let them and the patrons know that the test is available to the Davis community? If not, will there be plans to do so that people can actually take advantage of the test? That brings us to a topic of Healthy Davis Together. This is one of the most important topics, the relationship between the City of Davis and the University of California, Davis. We are working together in this process, and I asked the mayor of the City of Davis.
We are delighted to have the mayor of the City of Davis, Gloria Partida, who is joining us today, and she's able to explain to us what is Health Davis Together. And I believe that's much more than the testing that we are doing together with UC Davis. Thank you, Mayor, for being here.
Well, thank you for having me. I'm very pleased to be representing our partnership in this program with UC Davis, the city's partnership in the Healthy Davis Together. As you've said, this is a program which is a partnership between our two entities. And it has funding which comes from grants and also private funders. And there is quite a bit of money which is coming from these sources, and it allows us to provide a very robust program. And so, as you mentioned, the testing is part of that program, and it's probably the most important and visible part of the program, because it is trying to screen for asymptomatic carriers of the COVID virus. And for instance, in a 2-week period, they found 200 people, they identified 200 people with the COVID virus and who had no idea that they were ill and that were, traveling through the community and in their homes, spreading the virus. And so that is a very important piece of trying to contain the spread of COVID. But it's not the only part of the program. There's education, and there is support for PPE equipment, and there's also support for people who find that they are positive. And so if someone tests positive for COVID, oftentimes they need to isolate, they need to quarantine, but because of their living situation, perhaps they're students who have roommates, or they're part of a family, and it is very difficult for them to stay separated from the people that they are living with. So the program provides hotel spaces or spaces on campus for them to provide, for them to do their quarantining in. They also, there's also funds that are going to support people who are homeless or who are housing insecure in the community, because, this is a very vulnerable population, and having them in the streets of the community also is a danger to the rest of the community. Some of the other things that the program has done is it's provided incentives in the form of gift cards, for instance, to businesses, which does a dual, it dually supports businesses and also incentivizes people to go get testing. And they've also done things like for Halloween, for instance, they gave out these mini grants to students to provide alternative ways to celebrate together in a safe fashion. And so there were things like scavenger hunts or other very creative ways for people to continue to make connections with each other in safe ways. I think that a huge part of what they are trying to do is to work with people's behavior. And that's important because as time has dragged on, the fatigue of being socially distanced makes it very difficult for people to continue to do the things that they have to do to stay safe. It becomes very, very difficult. So there are many avenues that the program is taking to try to prevent the spread of COVID.
There is more about it, Davis, Healthy Davis Together later. We have some surprise for you. How accurate, Paul Jacobs asked this question, how accurate is the sewage COVID detection test? I have heard of reports that UC Davis sample specific domes building to trace it back at that level. Why aren't municipalities doing this statewide, nationwide, and worldwide? So I asked Heather about this topic.
Hi, Paul. Thanks for your question. I already answered the first couple of parts of your questions, part of Bryson's question, but the last part in terms of, why isn't this being done in municipalities throughout the country, throughout the world? And the answer is, it is being done. Wastewater-based epidemiology is being implemented as quickly as possible in many locations around the country and around the world. I just checked a dashboard put together by Dr. Colleen Naughton at UC Merced and Dr. Maureen Kinua at UC Davis, and they've identified 900 sites where wastewater-based epidemiology has been implemented in some capacity. So that could be as little as one sampling location that's being monitored periodically to hundreds of samplers being put in place in university campuses or in cities and are being monitored as often as daily. So there's a range of efforts that have been put in place. About more than 180 universities are engaged in this effort in 45 different countries, all indicated by this dashboard. But one of the challenges and why this isn't happening everywhere is that there is a lot of infrastructure that is not in place in order to do this kind of sampling. Wastewater-based epidemiology has been implemented in some other contexts. One of the prime examples of that is in monitoring the eradication of poliovirus and any resurgences that came back in certain cities. So the tool has been used to monitor other viruses in communities at that level, but not at the scale to monitor the trends of a global pandemic. So what we're doing here at UC Davis and at campuses all across the United States are trying to implement these programs to support our communities and the testing needs, and at the same time conducting research to understand the efficacy, to come up with the appropriate strategies for sampling, to develop the right sample processing protocols, the communication protocols, getting that information out to decision makers and out to the public for awareness. So we've made a ton of advancements. There's been a lot of collaboration since the beginning of the pandemic, of researchers coming together, sharing methods more quickly than we usually do to put this in place. But there's also a lot left unanswered. So as we see the light at the end of the tunnel with the vaccine coming into play, wastewater-based epidemiology will remain very helpful to monitor the trends of the virus at a community scale. We can even look, be sequencing environmental samples to look for different strains of the virus in these samples. There's a lot that can be learned to understand what is the potential for this tool to remain in place and to be expanded to support community efforts and community monitoring over a longer term.
Next, we have a series of questions. Richard, please take note. There are questions under the same umbrella of going forward.
With the new variant strain of COVID-19 being discovered in Europe, now being seen in California, are further precautions going to be taken at UC Davis to limit the spread? Since this new strain appears to be more contagious, will more frequent testing be required?
Hi, my name is Eva Pack, and I am a junior in the Biochemistry and Molecular Biology program at UC Davis. Would this test be able to detect the new COVID-19 variants that have been recently thought to come from South Africa and the UK? Thank you.
Okay, you need to stop sharing.
No, just 1 second. We're not there yet. There's 2 more questions in the same line.
Okay.
My name is Sudev Nambudiri, and I'm a junior in the General Biology program at UC Davis. My question is, what is the future roadmap for the development of the COVID test in 2021? In the next few months, will there be more of a focus on decreasing the amount of time it takes to get a result, or increasing the accuracy of the test?
1 more question. Just 1 second left.
Hi, everyone. My name is Paul Lusu. I'm an emeritus professor here at UC Davis at the Center for Immunology and Infectious Diseases. First, I want to say that I very much appreciate the superb efforts at establishing the large-scale coronavirus testing that is now benefiting the university and the City of Davis communities. My questions: if a person's saliva tests positive for the coronavirus, will there be a confirmatory test? In addition, what is the path or procedure for a confirmatory test? Thank you for the opportunity to ask these questions.
Thank you, Richard. I'm going to stop sharing here so that you can share from there your
Great.
segment on the going forward vaccination and new strains.
Great, thank you very much. So going forward, great questions. I'll try and address them in the short time we have available. One point to remember is that frequent testing is only part of the mitigation strategy. Just because you're tested, it doesn't mean you can go out and party. So testing alone does not control COVID. Wearing masks and limiting personal contacts do. Now, in terms of where we're going, we're trying to increase throughput. We're at 4,000 a day. We're ramping up to 8,000 tests per day. We think it's more than sensitive enough. Once we get up to 8,000, some people would like us to go higher than this. But one of the things we'd like to move is to expansion of testing to twice weekly. Clearly, if you're testing twice weekly, that disease curve that I showed, you're going to shorten that window where there may be infectious individuals walking around the community. We've expanded to Healthy Davis Together. We're helping the med school with their asymptomatic staff. We're reaching out to UC Merced and Yolo County. We're doing some of the assisting living facilities staff. We have a tremendous throughput. It's large, but unfortunately it is finite. And much as we'd like to help other communities, this is about all we can do at the moment. Going forward, thinking about alternative technologies, rapid antigen tests, I think, are increasingly going to play a role. You know, I'll be happy to return to my day job if there was a wonderful other test coming along. I don't think that's going to happen in the next 6-12 months, partly because we're so high throughput, we return the results within the next day, and we're cheap. Rapid antigen tests are about the same price. They are faster, but they're comparable. And then the importance of testing after vaccination. This is another question I'll address in a moment, and the impact of the new strains. So just a point, we've already heard about Healthy Davis Together and all the great things they're doing. We're just one part of that. The workflow is that middle bit, it's the accessioning. That's the rate limiting step. It's not the qPCR machines. If we could make that more efficient, we'd be able to go higher on the throughput. Importance of testing after vaccination. If you get vaccinated, do you need to be tested? And the answer is an emphatic yes. The efficacy data for vaccine is based on symptoms, not nucleic acid tests. And the reports of 90 to 95 efficacy relate to the prevention of disease. Now, you've been hearing through this talk, 40% or more of infections are asymptomatic. yet there's very high viral load in some of those individuals. And there really is, at the moment, there is no data to say whether vaccinated individuals can become infected or be infectious to others. So it is extremely important that we continue to be tested after vaccination, and we'll be monitoring vaccination records along with the test results. It's also very important to monitor for variants of the virus that potentially escape the protection of vaccines, and such as the potential strain out of South Africa that's in the press at the moment. And then the impact, there's a lot to digest here, but I know there's a lot of interest. And I would encourage you to go to that website up the top, thenextstrain.org. There's a tremendous amount of information there, particularly put there by Emma Hogcroft. And so SARS is continually evolving, though fortunately relatively slowly in comparison to other viruses. There are 3 new strains of current concern, the so-called B117 UK strain, the B1351, the South African strain, and one that's just come into the press, though, that was identified in Japan, but with travelers that come from Brazil. These are all potentially more contagious because of an amino acid substitution in position 501. And I've got a model on the right here from Emma Hogcroft's site. You can see it's a mutation that affects the binding interface to the ACE2 receptor shown in green. And this is thought to why it's one possible reason why it's more contagious, that once it's inside the cell, it doesn't necessarily cause more severe symptoms, though it's possibly replicating more in the respiratory tract and therefore also aiding contagion. It is in San Diego. It is going to spread throughout the country if the European data is any guide. And I'm sorry to say, it's going to get worse. If you can imagine, if you look at LA, it's going to get worse because of the more contagious strains. We've got to be very, very careful about the amount of contacts and wearing masks. The B117 strain was identified as a dropout of the S gene. That is, there is a deletion that happened to be in the priming site that was used to detect the S gene in the viral tests. And what they noticed was 2 of the other viral genes they were trying to detect, they were high, but there was no S gene. And it's not unique to this strain. About 50% of viruses sequenced were the B117, but there was another 45% that weren't. The Davis test, it relies on detecting the N1 and the N2. Therefore, our test is not affected by mutations in these viruses. We do, however, monitor for sequences for variants that could affect our test. We're also, though, developing a high throughput SNP assay that we can run on positive samples to distinguish and detect the viral strain. So that's what we're doing here. Again, we're taking technology from the agricultural biotech industry. They run millions of data points a day. Sequencing is too slow. It's very important to determine variations, but we can look at thousands of positive samples a day, and we're collaborating with LGC at the moment to develop this test. We're also preparing to sequence positives, but that'll be much more lower throughput. There's a national effort called SPHERES, run by the CDC, and COVIDNet, run by the California Department of Public Health, which we are participating in. And as Heather Bishoff just said, they're monitoring wastewater, and we're collaborating with them to see whether our SNP assay is going to allow much higher throughput and much faster, because you can turn around a test in an hour and a half. So that is, I hope, taking care of your questions.
Yes, and you already answered also one question that is just coming through YouTube from Sarah Ventinini. She was asking about the possibility of the virus not being detected because of the change, the variation. Let me look at this thing here.
Our test will detect the variants,
right,
regardless, but we don't distinguish them. Therefore, we need the secondary test. I would like to say, regarding that question about worry of going into the ark, the safety people did a tremendous amount of work looking at air exchanges And that is a very large space, and there's very frequent air exchange from the outside. It's about as good as being outside without standing in the rain.
Very good indeed. Okay, so let's continue here. Let me share the what I call the segment 4. I separate this thing in various segments because it's too big for the PowerPoint to handle. Segment 4 now, and the first of all, I would like to thank the officials from these different municipalities. The listing here is shorter because I did 2 days ago. We have now 206 officials from 76 municipalities throughout the state of California. That shows how they are interested in this topic, including the city of Danville. The city of Danville is the home of the Captain Sully Sullenberg. I'm still waiting for him to sign my book. I hope he'll have an opportunity sometime later in the future. So we have all these municipalities here represented by the elected officials, city mayors, and the city managers and the staff. And I asked the mayor about
we
that.
are very happy today to have officials from many municipalities in the state attending this symposium. So what kind of message you would give to them?
Oh, I would say to please just continue to get your messaging out. I think that communication and messaging is very important. I think trying to be as creative in getting that message out. There are large populations that are much more difficult to reach. And I think that, one of the things that we are doing with Healthy Davis Together is enlisting every single community leader that we can. So we reach out to congregations because congregations have very close-knit communities, and it's a great way to get information out. So we have congregations, we have community groups, we have nonprofits, and the Latinx community is one of the hardest hit communities with this pandemic. And so trying to reach out to that community has been particularly difficult. But I think that, again, the communication and the messaging are the most important things that we can do, and get tested. And just, I think it's really important that we encourage people to continue to get tested. I think very often people don't know why they should get tested if they're not sick. But I think making sure that people understand that there are many, many people out there who are asymptomatic.
Thank you, Mayor. We continue now. We are having the great pleasure of introducing the Chief of the Division of Infectious Diseases and Director of Hospital Epidemiology and Infection Control at UC Davis Medical Center, Dr. Stuart Cohen. He conducts clinical vaccine trials, clinical trials for the treatment of COVID-19. His research also includes serological testing to evaluate utility and epidemiology, and he takes care of hospital infections control related to SARS-CoV-2 infection. As you imagine, he's a very, very busy man, and we are very glad, very appreciative of the opportunity that he's given to us to be here to talk about vaccines and other things related to that. Dr. Cohen, a great pleasure to have you here today.
Thank you, Dr. Leal, and thank you everybody for sticking around. It's feeling like it's getting a little bit late. So I have a handful of slides, and then I'm assuming there will be some questions. So we will start with the next slide. So I think everyone is well aware of all this. This is a pretty sophisticated group, but I guess I wanted to point out two quick things, or maybe three based on this. First, as everyone is well aware, the spike protein is what binds the SARS-CoV-2 to the ACE receptors in the respiratory tree and allows people to become infected, and that the target of the host immunity, at least the humoral immunity in the host, is the spike protein by developing neutralizing antibodies. This has been, when you hear about monoclonal antibodies being used, they're all monoclonal antibodies that target different portions of the spike protein. And that's also the target of all the vaccines. The one other thing to just sort of touch on that Dr. Mitchellmoore already has addressed is RNA viruses. The way that they develop genetic diversity is they mutate. And genetic diversity is important in their survival within the environment. And so it is not surprising that this B117 mutant showed up or any of the other ones. And I can guarantee you that it's everywhere in the United States, just like the infection was everywhere in the United States when people were worried about travel. So I think that this is just how RNA viruses work. It's not a surprise, and it will end up impacting ultimately how long vaccines are last and whether we're going to be doing vaccines for COVID like we do vaccines for influenza, where we have to take them every year or every 2 years or every 3 years. That's yet to be determined. The next slide just points out the whole herd immunity idea. So the point of this slide, which is old, it's from September, I believe. And so some of the numbers may have changed, and I'll touch on that in a minute. But the idea is that you want to know what percentage of the population needs to be immune in order to stop transmission of a particular pathogen. And so at the top of the slide, in I guess it's purple, is measles. And measles has a basic reproductive number, or an R0, of about 14 to 15 people infected for every person. So in order to have herd immunity to control measles, you need to have over 90% of the population immune. With SARS-CoV-2, which is sort of in the middle and is in black, the R0 is thought to be somewhere around 2. I think with these new spike mutant viruses, it's probably over 3. And so the numbers needed to be to provide immunity and herd immunity to the point that transmission stops, you're looking at a minimum of 60%, probably somewhere in the range of 70, 75%, maybe a little bit higher. So the only way to get this done without an unmeasurable and inconceivable amount of mortality is via vaccine. So the next slide just points out the 3 different strategies that have been used to develop vaccines to the coronavirus. So the bottom is the mRNA vaccines. Those are the 2 vaccines that have been FDA approved, and I'll talk about them in a little bit more detail in a minute. But the mRNA codes for the spike protein, and the spike protein is what the immune system targets. The in between is using adenoviral vectors. The AstraZeneca vaccine, which was also worked on at Oxford, has been approved in Great Britain, and it's been approved in India. And then there's a second vaccine Janssen makes that's actually the only single dose vaccine that is also based on adenovirus vectors. So once again, these are replication incompetent vectors. They just serve as a way of bringing virus into cell or bringing nucleic acid into cells, and then it produces spike protein. And then the top is the somewhat more traditional strategy of making a subunit vaccine. And this is what's being done by Novavax and Sanofi. And the Novavax trial is the one that we have ongoing right now. And I'll talk a little bit more about that in a minute. The next slide just points out that all these vaccines are delivered in some form of a nanoparticle. So the mRNA vaccines use liposomes as a way of delivering RNA to the host, since you can't really give naked RNA to any person and expect that to survive. And this is where allergic reactions occur. If they do, it's typically the polyethylene glycol that's present in this lipid and phospholipid envelope. There are a bunch of other strategies that people have used to try to develop nanoparticles, but most of them tend to be somewhat based on lipids. The next slide actually is a cartoon that is suggestive of how things work for the Moderna and Pfizer BioNTech vaccine. The lipid nanoparticle contains the messenger RNA. Because you have to preserve this nanoparticle and preserve the RNA, they have to be stored at lower temperatures. The Pfizer vaccine a little bit more concerning at -70 for some rural areas and things like that. But in larger urban areas, there are -70 freezers and it does not seem to be a problem. The Moderna vaccine is at -20, which is a kind of freezer that you would have at home. So I think that's what is being injected. And when these are injected, within the muscle cells and within some of the macrophages in the area where the vaccine is infected, these nanoparticles are taken up, the RNA is released, and then the spike protein is made and displayed on the surface of these cells, as well as there may be some free virus released. You can go, well, you could stay there. And so I think that that's how the immune response is developed. Once the RNA makes the spike proteins, the RNA is destroyed. This is not viable virus. This is not detectable virus. Nobody gets infected from getting these vaccines. Next slide then. So the Novavax vaccine is a subunit vaccine, and this takes advantage of baculovirus, which is where the molecular material for the spike protein is cloned into, and then they use moth cells to produce the actual spike proteins that are then put on a commercial matrix so that you can see it comes out as these spikes sticking off this cylindrical object. And then they actually add a lipid adjuvant, which allows for an improved immune response, and it allows you to use about a fifth of the antigen in order to get a similar effective antibody response. And the phase 1/2 trial also looked a little bit at cell-mediated immunity and T cell response, which also appears to be stimulated. So those are the current vaccines. The next slide. So this was already pointed out by Dr. Mitchellmoore, so I am not going to spend much time on it. But I will point out that this has been a concern as to whether the vaccines would be effective against these particular strains. There are not a lot of data, but my last slide actually shows some information that is in a bioarchive preprint that looks at the black shows you the normal wild type 501, and then the red is the mutant 501 amino acid. And these are plaque reduction assays in laboratories and looking at neutralizing antibody titers. The diamonds are 2 weeks after the second dose, and then the circles are 4 weeks after the second dose. And what you can see is that if you sort of follow where the diamonds are and follow where the circles are, they tend to go, the circles tend to go up. The diamonds tend to line up, as do the circles, for both the wild type and the mutant virus. So it appears, at least with the Pfizer BioNTech vaccine, that these mutants don't appear to affect its ability to produce neutralizing antibodies. And so that should be a positive thing. And I think it all the further emphasizes getting people vaccinated and getting this thing under control, because every time this virus is passed it in another human, there's potential for more mutant strains being developed, right? This is, the more people infected, the more likely you're going to see more diversity and more difficulty putting the genie back in the bottle. And I think that's my last slide. And so I will stop and take any questions that you may have.
Thank you very much. There is one question here next.
My question is, why do we need 2 doses of the vaccine? What if we only got 1 dose? How protected would we be?
Yeah.
Yes, go ahead.
Go ahead. No, go ahead, Walter.
No, it's a very common question, and also another type of question that came very often is that can we take 1 dose from one and another dose from another one? Which
yeah.
Question.
So I can answer both. So so the optimal titer of neutralizing antibodies is developed after 2 doses. So the second dose functions as a booster on top of the first. If you look at the number of people who have symptomatic COVID-19, approximately 10 to 14 days after the first dose of either the Pfizer or the Moderna vaccine, the curves markedly diverge between those who receive placebo and those who receive vaccine, and the vaccine group is pretty flat. So I think you develop pretty good immunity after the first dose. The titers are not as high and the durability is not as clear. And so the second dose functions as a booster, which allows for higher antibody titers and presumably prolonged immunity based on that. The vaccines are hypothetically not interchangeable. The goal is to get everybody the same second dose as they had the first dose. And when you get vaccinated, you get a card that tells you what you received so that you know what you need to receive the second time. If somebody received the Pfizer vaccine as their first dose and the only option for getting another dose in a timely manner was getting a Moderna vaccine, I would not stop somebody from getting the Moderna vaccine. There are no data on that, but based on how these particles are built and what they target, it doesn't appear that they would be that much different. But the data will show, because this is going to happen. There's no way it's not going to pop up in one circumstance or another.
Very good. Very good information there. Mary Jean Allery asked, what are the absolute contraindications for the COVID vaccine? What are some considerations for taking the COVID vaccine? Examples, food allergy, history of prior anaphylaxis. Another question that relates, there's three questions related that I wanted to pose to you today. My son is 17, carries an EpiPen, and he has a rare neurological genetic disorder. He also has uncontrollable epilepsy. I am concerned about him taking the vaccine, particularly with his current medication. Are you testing the vaccine on people like him? And another question, the same line, is I have had two young adult human-friendly decline the vaccine due to unknown effect the vaccine might have on their reproductive health. One has since developed COVID-19 and is moderately ill. What's known about the effect of reproductive health of having the virus versus the vaccine? So these are all questions in the same line, and I appreciate if you address them.
Yeah, they're great questions. Can you go back one slide so I can actually remember what they are? I'm getting old and tired. First, as far as absolute contraindications for the vaccine, having had an allergic reaction to the vaccine or polyethylene glycol would be an absolute contraindication. As far as considerations like food allergies or prior anaphylaxis. So the way that we have been delivering vaccine in both clinical trials and now that we are vaccinating our healthcare workers, everyone has to sit and wait for at least 15 minutes after their vaccine to be monitored for allergic reactions and anaphylaxis. The patients who have had a history of prior anaphylaxis are generally monitored for 30 minutes and longer. And there have been anaphylactic reactions. You know, one of the things that's difficult is when you see a report of an anaphylaxis circumstance that occurred with the vaccine, there's no denominator attached to it. And so when these rare events occur, you have to put it in the context of how many people have been receiving the vaccine. So we are trying to get it out to other people. Now, if you were participating in a clinical trial, you'd be excluded if you had a bone marrow transplant or had a kidney transplant or were actively on chemotherapy or had HIV infection with a very low CD4 count. All those things are, and for that matter, when I get to the reproductive health question, pregnancy is also a contraindication to participating in trials. But I think that's going to be different when it comes to actually vaccinating people. Now, for the second scenario, so only the Pfizer vaccine tested anybody under 18 years of age. So if you just moved your son's age up to 19 or 20, where he would be eligible for any vaccine, I think the idea that somebody's on Lamictal should not dissuade them from taking vaccine, and he would not be excluded from a clinical trial based on having underlying epilepsy. And then the next comment was the one about reproductive health. So first off, there's no evidence that it affects reproductive health at all. These are not live vaccines. So they are safe to be used in pregnancy, even though they have not been tested in pregnancy. Based on everything we know about every other vaccine, we have been able to give vaccines to pregnant women. They get influenza vaccines every year, even when pregnant. And so if you look at the potential complications to the mother and potentially to the baby, if mom gets COVID, the vaccine is a much better option. And our obstetricians are enthusiastic about vaccinating their people. But this is a circumstance where I think the woman has to talk with their obstetrician or their gynecologist and clarify what she or he has to say about her risks. Personally, I would not dissuade anybody from getting vaccinated.
Very good. I guess a general theme is that immunocompromised people also would be included in your definition, right?
Yeah, right. So when you look at how CDC and how the states have sort of ranked people for getting vaccine, in that so-called 1C group are people over age 65, but they're also people with underlying illnesses. And so, again, I think if you had an oncologic problem, I think the decision should be made in combination with the oncologist or with your transplant doc or something like that. But if the advice, if they come to me and ask for advice, I think it's safe to give these folks vaccine, and I think the disease is way worse than the vaccine.
Thank you very much. We have, if you wait a little bit, I have more question for you. We have another guest here. I'll let her introduce herself.
I'm Mary Krogan, Provost and Executive Vice Chancellor at UC Davis. I've spent more than 30 years working at the University of California, first as a professor and researcher in family medicine, OBGYN, and epidemiology at the UCSF School of Medicine, and later as Executive Director of the Research Grants Program Office at the UC Office of the President, and finally as Chair of the UC Academic Senate. One of my most important qualifications for being provost is that I'm a proud Aggie.
Thank you very much for making the time to be here today. I know that we're busy with some meetings and could not confirm, but now I'm glad that you are here. Thank you.
Thank you for having me. Pleasure to be here.
All right, we have one question for you.
Hi, my name's Alison Van Eenenham, and I'm a faculty member in the Department of Animal Science at UC Davis. And I have a question about where UCD faculty sit in regards to priority for a vaccine, given that we are not K through 12 teachers. Thank you.
So the priority for higher education is actually being reevaluated over the last 2 weeks. And the state of California is considering moving higher education up to the same level as K-12 education. I think it's got a very high likelihood that it will be approved, at which point faculty, staff, and students in higher education settings in California will be at the same tier, which is tier 2, as K-12 educators. So we're hopeful that will take place.
Yeah, that's very important because people would think that right now we are doing remote teaching, but we are planning to, and we'll go back to that point in a minute, we are planning to go back to in-person class in the fall. So that's need to be moved up. So there are more questions for you. And let me see here. These questions, a bunch of questions related, and will UC Davis offer COVID vaccination on campus to members of the campus community?
So first and foremost, people tend to get their vaccines either from their healthcare provider or their employer if vaccination is required as part of their employment. Because the vaccine is under an emergency use authorization, we are actually not able to require it at this time of UC Davis employees. So it's very different than UC Davis Health employees, which Dr. Cohen is part of. So at this point, we are not able to require vaccination. There are very limited vaccines available in California right now. Our hope is that that will pick back up again in the next week or two, and there will be more vaccines available. We've actually made an application as a campus to the state of California with the hope of being able to open a vaccine site on campus. And we would do it in parallel to what you very ably showed, Dr. Leal, in your videotape of the Activities and Recreation Center asymptomatic testing site. We would do something similar there on campus for UC Davis affiliates. Given that our students actually do receive their healthcare through our student services, they would be vaccinated by the university in either case. We also spoke earlier this evening about Healthy Davis Together. You had Mayor Partita as part of this. And our hope is not only to be able to stand up a vaccination center for the campus, but to be able to have a vaccination center for the city as well. But again, as I said, this is dependent upon being able to receive sufficient vaccines from the state.
So before I go to the second question, I would like, since the providers are going to the one to deliver the vaccine, let me ask you, Dr. Stuart Cohen, about the patients that are under the care of the UC Medical Center. I understand that UC Medical Center already started giving to the normal population, not the medical personnel, but now already started with the population, maybe at a higher age level of people, some underlying condition. Can you explain it to the general public what's going on at UC Medical Center now in terms of vaccine?
Yeah, so we started yesterday with people 75 years and older who get their care at UCD, and it went reasonably well. I think there were about 70 people yesterday. I think it's about 100 and some today. I'm not exactly sure the full numbers, but we're moving along, and you know, we mimicked at the medical center what you folks on the Davis campus do for testing, and we use the same strategy as Dr. Cohen was mentioning for actually delivering vaccine. And so we have part of an area called the Mind Institute that's set up with a big auditorium, and we have people spaced out and walk in and get vaccinated. You know, we stood up a vaccine program for our healthcare workers. We vaccinated over 10,000 people in a handful of weeks. And so it's a doable thing. And once you've sort of figured out the infrastructure, it works well. And similar to what you're talking about with the Davis community, we are trying to support the community in Sacramento. We're vaccinating some of the healthcare workers that work over at the county clinics. And there are several other community partners where we're going to be providing vaccine as well. So I think, you know, the faster we roll things out, the better everyone is going to be, and the faster things move forward. And I would emphasize that it's hard to make something mandatory when it isn't fully FDA approved. We make flu vaccine mandatory for all our healthcare workers. We all have little dots on our badges that demonstrate that we've been vaccinated, and we talked about doing that with the COVID vaccines. It just didn't seem like it was something that we would viably do But as everyone has pointed out, if you want to get back to face-to-face classrooms and not have to be masking and putting people in corners of rooms, this is the way to do it. And as also was mentioned in the prior commentary with testing, this is why we still need to do testing, right? We still don't know whether they prevent infection. And even if they do, they're only 95% effective. And I hate to use the term only, but it's not perfect. And so there's still going to be people who potentially can be infected. And so there's still potential for transmission. And until the entire population, until you reach herd immunity, these little pockets are still potentially risks for people.
Thank you very much. That's very important information. Let me go to the second question to the provost. Our government and the private sector have struggled to have COVID testing work as rapidly and seamlessly as the UC Davis COVID testing. Given similar struggles on the part of the former to roll out vaccination, doesn't it make sense to have UCD administer them to the campus community? You mentioned about the vaccine not being approved by FDA, so probably that's a block road right there, right?
Well, I answered all three questions at the first time. But just to reiterate a bit, we would love to have the opportunity to administer the vaccine on a larger scale to the campus community and with the city of Davis in partnership with Healthy Davis together. The issue is that we need to be approved for receiving the additional vaccine doses. So as we've said, they're in limited supply at this point. It may be that some of the logistics of getting the vaccine distributed throughout the state and really throughout the nation will be addressed in the next few weeks. And I have faith that we'll be in better shape in a month to 6 weeks from now. So it may just be slow at this point, but we should be able to accomplish this.
What about this question here? When will graduate students who are teaching assistants be eligible for the vaccine? And also, is there any chance that UC Davis faculty will have any priority in vaccines? You talk a little bit about it. We are also being the tiers of K-12. When will graduate students be vaccinated? Basically the same question. So there are many questions in the same line. Could you please address it?
So two aspects of that. We are the healthcare provider for our students, even if they have insurance coverage still through their parents up to 26 years of age. UC Davis, through Student Health and Counseling Services, is a healthcare provider for all of our students. So that mechanism of standing up a vaccine clinic for our students is much more straightforward. But again, we need to have the vaccines in hand. With regard to the faculty and graduate students who are TAs, either way, they would come under the higher education rubric, which, as I said earlier, is being moved up hopefully in the tier structure for the state of California. So whether for a graduate student per se, they would either get vaccinated as a healthcare provider being UC Davis, or they would get vaccinated under their teaching responsibilities under higher education.
Very good. I would like to move here to the next segment. I may have a technical difficulty here, but I'll solve that momentarily. So that's the next segment. That's all right. So let's see what I can do here. There was no glitch so far, so that we have a chance to have a glitch now. So with that one, I'm not going to resolve that immediately. So I'm not going to try to dwell on that time. So let me introduce, without the slides, one surprise that we have today. We have been talking about Healthy Davis together. And now we have the opportunity to welcome the person that spearheaded that program. And it's a very successful program. I've been here at the University of California for 20 years, And I can tell that there were days that the relationship between the university and the city was not the best. And now we are enjoying a very fruitful relationship with the city of Davis through this program. It's a model that the other cities could do the same thing. And it's a very good example of what the land-grant university could do to solve local problems. So the person spearhead that program is Dr. Bradley Pollock. He's a professor and the chair of the Department of Public Health Science and Associated Dean for Public Health Science at UC Davis Medical School. Thank you very much for making the time to be here. I know that's late, but we had so much to discuss about Health Days together. Thank you. You have to open the microphone. Microphone is off.
No problem. My pleasure, Walter. Thanks for inviting me.
All right, so we have a question here that comes from Davis Enterprise from Caleb Hampton. What insights into expanded testing accessibility in other communities can be gained from the saliva-based testing at UCD Health Davis together?
Yeah, let me, and again, Richard, of course, has been the steward of the technology along with a whole cast of other collaborators. I think we're in a very unique position. I think there've been very few, I've not actually heard of any attempts to do this sort of massive asymptomatic community screening in a real community on a large scale nationally. And we've actually presented this to the CDC and other federal groups there. They're very interested in what we've done here. One of the goals of the Healthy Davis Together project is to actually create the rule book or the playbook for other communities. And so, of course, the technology and the bold introduction of a ag biotech solution that Richard had come up with early on is incredible. I think the other things that we've learned are actually the challenges of trying to implement this in a community setting. And I'll just point out that we've only opened up community testing in the last month, a little bit more than a month. And it looks from our statistics now that we may have about half the community that has actually at least been tested once. Of course, the idea is to have people tested on a regular basis, weekly, and perhaps going to twice a week. So we still have some challenges. I know Mayor Partida has been a phenomenal advocate. As you pointed out, Walter, this has been an incredible partnership to be able to work hand in hand with the city and the university together to sort of move things forward. And we've been working jointly on trying to get messaging out. And one of the problems we've had, of course, the challenges is you can't invite people to a resource that's not there. And it took us a while to go from testing our students to moving to larger groups, employees, and now to the whole community. So as I said, we just opened this thing up a little over a month ago, and we've then started getting the word out to try to get more people in the community tested. So obviously, the larger the proportion of people tested, the safer the community is overall. But our intent is to try to generalize what we've done to help other communities. And of course, college communities may be unique, but there are aspects of the Healthy Davis Together program that would be sort of compartmentalized that would also be generalizable to non-university communities as well.
The second part of the question from Caleb is how much government investment would it take for similar testing to be made freely available at every CVS in the country within the next few months? Is something like that realistic? Does the technology mean that in more university community partnerships like the one with UC Davis in Davis is a more likely model for expanding this kind of testing?
Well, that's a great question. Richard might be able to answer that more efficiently. LGC, which developed the platform that we're using, the PCR platform, in fact, they're a British company. And my understanding is when we were talking to them early summer, that they've actually rolled this out for the National Health Service in the UK. So clearly they have some intents. And of course, the Gates Foundation has supported an even larger throughput platform. So not a saliva-based test, but clearly this is scalable, but this is a first demonstration of use in a large community setting like this. So costs would have to be considered. It is clear from this pandemic that we were caught without the ability to do adequate testing. If you recall back to the beginning of the pandemic, the CDC was releasing 20 test kits at a time to large academic health centers, to be able to test 20 patients. And again, it was a novel virus, so we didn't have the kits available. But I think for the next pandemic after this one, we will certainly be in better shape. We'll have more infrastructure. We know how to do things better. And I think the testing part of this and looking at higher throughput platforms that are less expensive. I know that we talked a little bit about this with antigen testing, but I also think there's a strong case to be made for developing at-home tests, like the pregnancy tests. And there are a couple of kits that are out now to do that, where you wouldn't necessarily have to rely as much on central testing, which is very cost efficient the way we've set it up. But obviously, if people had a very simple spit test to do in their homes every in the morning, they just spit into the thing, dip the stick in there, and if it lights up positive, they stay home. They don't get out in circulation at all. That would be optimal. I know Michael Minna has pushed that. So I think we'll have to look at in the future about how we best deploy different testing technology for these sorts of pandemics. But the technology's definitely made this much more feasible now to do on a large population basis.
So what we refer to is different from what the UC San Diego has launched now, that they have the vending machine, but just for you to take the kit instead of going, for example, to the ARC to do the test, you just get in the vending machine there. What they're doing is one step ahead of that, where you get this machine and then you test yourself. That would be ideal.
Yeah, well, actually, to do it without a machine is even more ideal. That is to just have a very simple dipstick where you could do this at home and find out without having to do any technology other than the reagents that are on the stick itself. And there's actually, I think there've been 2 EUA approvals for those kinds of test kits. Still a question about whether they're intended for symptomatic versus asymptomatic type testing. But ultimately, I think that would be the best way to be able to handle things like this. But it'll probably be a combination in the future.
There is this question that was asked, you have been asked this question in national TV with Stephanie Rule, and she has a rule that only good people are there. So the program was very fruitful and she was highlighted that program there. Here's the question.
Less than $10 a unit. People can get in and out to get the test and they get the results within 48 hours. Could this have been done on a national scale?
So it's similar to the question of Caleb Hampton, but do you have an updated version of your response to that question?
Not much updated. I guess the answer is it would depend. And I just will say this, that we've had discussions with President-elect Biden's COVID-19 transition team leader just in the last couple of weeks, including a discussion last week, to try to, because she was very aware of what we had done, and to try to see if we could generalize this. I mean, I think the testing platform that we have in place has really been a centerpiece of what we've done to do community-wide testing, but the trick is to not only have that available, actually to link this to other things, which include possibly with people that light up as being positive, to try to get them into monoclonal antibody therapy, which has been vastly underutilized in this country, or to sort of streamline them getting into vaccination faster, perhaps. But I think, is it feasible? It's feasible. But again, obviously it's worked quite well in the ag biotech world for plant pathogens. It seems to be working well for us here. And again, UK is doing this. And as I said, I forgot the name of the machine that Gates has been supporting, but it has even probably half an order of magnitude more capacity to do this. But there's, I want to point out that the unit costs here, which are very reasonable, don't really cover the acquisition costs. And all that infrastructure we put into place to have those testing stations set up, the personnel that are there, how things are being handled, the acquisition costs still turn out, for us at least, they still turn out to be a substantial portion of our overall cost of doing this. The testing part of itself has been really reduced down, as Richard said, by an order of magnitude over some of the commercial tests. But again, getting the specimens is still the challenge, and then handling them is a challenge. We've gone to robotics now to increase our throughput. My understanding, Richard, you can confirm this, is the robots work most of the time, but there are still issues that have to be worked out. So again, scaling this up, probably not a bad idea now, But in terms of how we'd use this in the future for future pandemics, it still remains to be seen what we would do.
I have another question for you and Richard in a minute, but I also would like to ask Dr. Cohen about this important issue about the doses of the vaccine. The initial plan was to retain half of the vaccines and make sure that whoever got the first injection would get the second one. Now, with this thing spreading so rapidly, they change in nature and they say, okay, let's give everyone the first dose. Maybe we get the second one if we get enough. So what's your take in that, Dr. Cohen? As a medical doctor, we are not talk about the politics in here. We never talk about the politics in this symposium. We are interested in understanding, is that a good idea or is a bad idea? Because we have here on the one hand, we have a pandemic that's spreading very rapidly. And as you mentioned, the reproduction number probably is going to increase in 50%, going from 2 to 3. And now what we do about that, we own a subset of the population. So I would like to hear your opinion about that, Dr. Cohen.
Yeah, so when we were doing our initial vaccine planning, we were, even for healthcare workers, we were talking about holding back half the doses. And then as things started changing, we realized that, as has been mentioned, the vaccine is not plentiful. So there is some concern sometimes that there may not be second doses after you give up all your doses of vaccine. But I think you use what you got, you get as many people vaccinated. I think there have been a bunch of questions in the Q&A that I've been trying to answer to the best of my ability at 7 o'clock at night. And so I think the answer is, if it's a little bit late, it'll be fine. And we just got to get more people vaccinated. You get some immunity by 2 weeks after the first dose. We just got to get it done.
All right. So another question is that whether one should take the shingle vaccine and the COVID Moderna vaccine the same day. I don't think anyone should take anything to get Shingrix in the same day, right?
No, we don't want people to take, well, first of all, we don't want them taking any other vaccines for at least several weeks. We like them to not have had another vaccine for 4 weeks prior to getting a COVID vaccine. I think it's a mistake to do them all together. As you sort of are alluding to, Shingrix is one that many people have significant reactions to. I think that it's based on similar types of adjuvants that are included in the nanoparticle of the mRNA vaccines, and also as adjuvants, for example, for the subunit vaccines. So I think you end up having much more severe reactions. I know that at least one of my family members that got Shingrix had temperature to 102.5 and shaking chills. I mean, it's not a joke. And same thing can happen with this COVID vaccine. So you don't want to back them up together.
But having said that about Shingrix, I would change the 1 day of Shingrix that I have after the vaccine with any of these 2 weeks that I spend in a hospital in Japan. So it's always a management problem, right? So the vaccine may have some side effect, and people complain about to get a sore arm about this vaccine. This is nothing compared what people are suffering with COVID-19. So that is always
right. This is exactly right. I think when you look at it, there are people on ventilators. We're not as in as rough shape as in Southern California, but approximately 10 to 15% of the people in our hospital right now have COVID-19. At places like UC Irvine It's probably half of their hospital has COVID-19, including significant numbers in ICUs. This disease is not a joke. And so a sore arm or a fever or a headache is a lot less of a headache than you're going to have if you're sitting on a ventilator for 2 months.
Exactly. Take that vaccine at your earliest opportunity. The Yolo County is asking people to register. The Davis Enterprise have an article yesterday asking people to go online there. And one of the questions that they ask is that would you be available if this vaccine became available for you within 30 minutes to come to our place? And if you are, maybe someone give up or someone doesn't show up, they have this slot empty and one can be called for that. So I also encourage, I read the article on the Davis Enterprise yesterday and immediately I registered, and I encourage everyone to do the same, not only here, but also in their own communities that are probably doing the same thing. So let me-
I think it's important to realize, right, that once you thaw these vaccines, you have to use them.
Yeah.
And so if somebody doesn't step up to get that other dose of vaccine, it gets tossed. It's a total waste. So that's why people are reaching out like that, and why you have to, right? It's like getting an organ transplant. You have to be ready to move when the organ is available. And that is, that's sort of how this plays out.
One thing about the article that I was not very happy with is that they say that they are being able to get more doses from the vial than is recommended by the Moderna in that case. So I'm not very happy with that. I don't like the idea of giving a dose that's under the amount. I don't know how they are doing that, but I hope they are not changing the specs because we need what was tested, right?
Yeah, and they're not changing the specs. There's just more in the vials than is necessary to give. For example, in a 5-dose vial, there's usually six doses worth of, and that gives people a little bit of a cushion when they're pulling it out. And if they waste some, they're still able to get the vaccine out of it. But if you're good about it, there's an extra dose, maybe 2 extra doses in some of the vials.
Oh, that's
just the way they're but no one's getting a half of a dose. Everyone's getting the same exact dose of vaccine.
I'm waiting for a long time and I want to get the full dose. Very good clarification, because maybe the vendor, to be at the safe side, they put a little bit booster there so that it's more than material. And if they carefully, they can take it instead of 5, they 6, which is a great thing. That's an increase in about 20% right there. Let me, the provost is there. So I have one more question here that maybe Richard or Brad can answer this question, one each. Can we do pooled testing of saliva samples? Example, 12 saliva samples for a classroom pooled for the cost of one test. And more importantly, can a non-medical volunteer be trained to collect saliva samples off-site? For example, in public school employee or volunteer to collect saliva samples at the school site if Davis Unified School District decides to have in-person class with COVID testing. Can both of you?
I can answer the second, but Richard, you want to take the first one?
Yeah, I'll take the first one. Pooled sampling is good if the limitation is in the throughput of your machine. The throughput on our machine is not limiting. And if you do a pool, you then have to look at the positive pool, break it apart. So it delays the turnaround of the test. And also pooling only works if the frequency, the prevalence is pretty low. If you get up much above 5%, and certainly if you're above 10%, it's useless. Every pool will be positive. The other thing is we run these very small reactions, and the cost of the reaction really isn't limiting. But because it's a small reaction, we do have a little bit of reduction in sensitivity. And I would be concerned if we went to pooling, we might lose a little bit of a critical level of sensitivity. So for our test, pooling is not the right way to go.
Good, and I'll take the second one. We're actually doing this. The second one is talking about non-medical volunteers. Actually, I hope people know that part of our Healthy Davis Together, we've extended testing at congregate facilities for the aging folks. That's skilled nursing facilities and assisted living for all of Yolo County. We're helping out. Now, they were required to do testing, but the frequency of testing was not quite optimal. And so we've volunteered to come in there. And in fact, we're training their employees now to be able to do the saliva collection. We also plan to use the same scheme when the Davis public schools open up. So we want to use that same thing of doing some central collection there from some of their personnel, which cuts our costs a lot. And I think the other option that we kind of entertained early on was the grab and go option. This was something that we thought about several months ago, which is even for our students to be able to do this perhaps in their dorm room and then bring the specimens in. We elected not to do that. And in fact, if we did do that, it would require a separate emergency use authorization review and so on. But I have to say in the last week, we've been re-entertaining that idea to be able to extend the testing capacity out beyond the current levels that we're doing. So it's theoretically possible, of course, but it's an excellent question. I mean, the idea here is to make testing as convenient, quick as possible. So as I said, we are doing this though in the congregate living facilities now in Yolo County.
Dr. Cohen, there is this question here from Tia Strong from Sacramento. She asks, will previous infections with COVID change the severity of the vaccine side effect?
Yeah, so I'm going to just speak anecdotally because people were not tested in the trials other than to determine that they weren't actively infected when they got vaccine. So no one got antibody tests before they were enrolled in trials. So we didn't know whether they were actively infected. Anecdotally, what my colleagues who send me emails about their side effects are commonly people who have been infected previously. And that's why we tell people, wait 90 days. I think there was one message in one question in the Q&A that asked that question. So we like 90 days. We know that the immunity lasts at least 3 months. I think there was a recent paper now that says 8 months. So it gives other people an opportunity to get vaccine. It allows for more people to be vaccinated while people who have already been infected have some immunity. And I think if you spread it out a little bit more, it seems less likely that you'll get a significant adverse reaction to the vaccine. I have not looked at this in a systematic way. This is supposed to be a scientific meeting. I'm the only guy that lives and works in Sacramento, so we're not as scientific as you guys are in Davis.
One question for you here from Chabra from Grand Bay. She asks, can a nurse who works on the COVID floor, who has been vaccinated with the Novavax vaccine according to protocol, brings the virus home and infect others?
Yeah, so first off, I just want to point out that we provide all the appropriate PPE, and just as Dr. Pollack was talking about unprepared for testing, we were totally unprepared with PPE as well, with personal protective equipment for healthcare workers, and people were being put at risk. But if you look at the healthcare workers who work constantly on a COVID floor, we have very few that have been infected. We have very few people who have been infected at work, but many of the people who've been infected at work have been infected by their colleagues. And there also are non-COVID floors where we've had more problems than on the COVID floors. So if somebody gets vaccine, regardless of which vaccine it is, they may still be infected and they therefore could still potentially transmit, which is why we're not stopping all the PPE once people are vaccinated within the healthcare system until we know that it actually prevents infection, not just disease. And so the same would occur at home, but that's occurring right now before they get vaccinated. And if they haven't infected them now, they're probably not going to infect them later.
So another question that comes from Jill from Fort Bragg. You talk about the immunocompromised people and people with underlying condition, but she has a specific question here. Is the vaccine safe for those who have had a diagnosis of Guillain-Barré syndrome? And also, she asked a second part of the question, do you have a better understanding as to why the virus causes some people to continue to experience symptoms longer after exposure, the long haulers issue? So these are two separate questions probably.
Yeah, neither of them are particularly easy, but I will do my best. So regarding Guillain-Barré, that is not listed as a contraindication to the vaccine. Guillain-Barré was raised to prevalent or to prominence during the swine flu pandemic back in the Jimmy Carter era in 1976, where the vaccine was associated with a higher rate of Guillain-Barré. At this point in time, we don't really see that as a big issue with influenza, but we do make that a contraindication to influenza vaccine. But there's no other vaccine where we ask people if they've had Guillain-Barré and where we make it a contraindication. So there's no reason to do it at this point with the COVID vaccines. Regarding the long haulers, that's a trickier question. I would like to point out, though, that if anyone has ever been really sick, whether you've had hepatitis or whether you've had some other serious illness where you were hospitalized for a couple of weeks, it takes a long time, right? You work out really hard to get yourself in shape, and it takes no time to get out of shape and to really fall apart. Not only that, people who get really sick in the hospital, they get significant damage to their lungs. That doesn't repair immediately either. So a lot of this stuff is just sort of natural history. I always use an example of one of my colleagues who got hepatitis A before routine vaccinations in one of the restaurants here in town. And he told me it took him almost a year to get back to where he really was. So I'm not surprised by this. I think the seriousness of the illness and the amount of energy that the host takes to get through it all makes it likely that these long haulers are there. This is my own thoughts about it. I may be proven wrong tomorrow, but I think that it makes sense. It's something that's really well known with almost any serious illness.
Can you talk a little bit about the Novavax clinical trial? I think you are targeting minorities because the other vaccine didn't have a chance to do that. And my question is, because now in the past it was quote unquote easier to get volunteers because there was no vaccine available. Now that there's a vaccine available, and if someone is going to go to a clinical trial, they might as well get the placebo because the trial has no meaning if it doesn't have the placebo. So do you find a little bit more difficult now to get volunteers for these trials, or things did not change?
So I was really nervous about it. This trial took a lot longer to get started than everyone thought it would take. And so when I got an email like the day after Christmas saying, if you want to start enrolling tomorrow, go ahead, it sort of came out of the blue. It was like, sit around and wait, and then all of a sudden it's bang, you got to do it tomorrow. And so I thought this is going to be a problem because the EUA is available and we want to target high-risk minority populations, but we also want to target older people, right? Because that's where the disease is most prevalent and causing most problems. And so we want to demonstrate the vaccine works in those groups. I think there's a plan that's already in place that if somebody gets called up and gets offered an EUA vaccine and they want to take it, The study has already said that they'll unblind them. And if they've not received vaccine, that they can go ahead and get EUA vaccine. It will end up making the trial more problematic, but the goal is really to get people vaccinated and determine whether the vaccine works. It's a 2-to-1 randomization, so 2/3 of the people are going to get vaccine, which is a little bit different than some of the other vaccine trials, which were 50-50s. And we've had a lot of hits. So we've actually shut down our email site through UC Davis. I'm still getting emails through Novavax. We have over 2,000 people who have expressed interest. We're trying to enroll somewhere north of 200 people, depending on how it enrolls nationally. And it's a 30,000 person trial. It's about a quarter enrolled already. So I think there's still been a lot of interest. I think because there's a surge right now, people have been more enthused about getting vaccinated, and they see this as a route of getting it more quickly. If everything is organized, as Dr. Crowan brought up before, and there's now a lot more vaccine available, I think it will potentially affect the trial, but that's how it goes. And so I thought it would be a headache. It's really not been. The biggest problem right now has been that we need space to be able to get everybody through in and out. And there are still people from the Pfizer trial getting vaccines now, all the placebo people. And so we don't have as much space to move people through as rapidly as we'd like.
So all the medical personnel that want to take the vaccine at the medical center already got that. Is that right? All students also, medical students. Is that
medical
correct?
Students have, and residents have, and all the people that actually work in the hospital or who have patient contact will have been vaccinated if they want.
And the nursing students also.
Right.
Okay, so there's nothing we can do at this moment if someone decides that I don't want to take this vaccine. And there are different reasons for people to think about that, but there's a large majority of people wanting the vaccine, and they want this dose that people do not want to be available to them immediately.
Well, so we are talking about putting together what are being called vaccine concierges. I don't make up the terms, and so I think that we're trying to get some education out and trying to get people interested in being vaccinated, but you can't force people to do it. And I think that ultimately people figure out what's going on. Some of the people I know that have not been vaccinated are pregnant, and because I can't guarantee safety, which I cannot, they want to wait. And so I get it. I understand. Some people just are adverse to vaccines. The one thing I do want to bring up is that this has been a condensed timeline since the clinical trials were started, but this is actually how a vaccine trial works. You enroll a lot of people, the disease is relatively short incubation period, and you identify people, and by 3 months, you know whether the vaccine works or not. The reason that these studies are carried out longer, and all these studies are scheduled to go for 2 years, is to look for any long-term complications. That's what we don't know yet because we haven't had long-term follow-up for anybody. And the goal would be to have people stay in the trials through the 2 years. Even if they get vaccinated, there'll be a little bit of a staggering of groups so that maybe some information could be gleaned from that. The vaccines for coronaviruses were really first developed, and the ideas were developed that the spike protein was the target back in 2003 when the first SARS virus was circulating and people were developing vaccine to SARS-CoV-1. That was also the first time people talked about using mRNA vaccines. So this is really 17 years in the making. It just, once the data were available, it didn't take that long to develop an RNA vaccine once you know what the sequence of the virus is. And so things moved faster because the background was already there and things moved along. No one is surprised now that, about a week after the first case, we already knew the entire gene sequence of the virus. I started practicing infectious diseases when we didn't even know what was causing AIDS. And it took 2 years to identify that virus. So science moves faster now. This stuff, it isn't growing vaccine in eggs anymore. This is moving along. And so I don't think that people should fear that this was rushed along or done or shortcuts were taken. The ground was already plowed and all they had to do was plant the crops to get this moving.
But there are people out there that asking this question. We have many of this question here. Oh, this vaccine is going to become a part of my DNA. I don't know how to explain it's possible for RNA to be incorporated DNA. And as you point out, these RNA is so unstable that you have to protect them with this particle to be able to then to arrive to the cell. And they are immediately destroyed. And people need to understand and to believe what the science says, because that's not what we know.
Right.
Another issue that they bring about is that, these use human kidney cells, and maybe are they referred at some point in the process that someone use a hex cells. It doesn't necessarily mean that this came from an unborn baby that someone borrowed that cell. So this is not the kind of science. Hex cells can be from a volunteer, from someone. It doesn't necessarily mean. So this kind of disinformation makes people more averse to the vaccine when that's not there's no foundation in the science, right, Dr. Quinn?
Yeah, I agree. I was in clinic yesterday. I think yesterday. Today's Wednesday, right? So I was in clinic yesterday, and I had one of my patients. This is an HIV clinic, and one of my patients said, "I've heard that." First, she asked me whether I'd been vaccinated, and I told her I had been, and she said, "So you thought it was safe?" And I said, "Yes." And she said, I've heard that there's HIV virus in these COVID vaccines." Right. So this kind of stuff. What can you do when this kind of stuff gets circulated around? It's very difficult. And that's why these people that are trying to work with some of our unenthusiastic employees are basically saying, "Look, I rolled up my sleeve. I took this vaccine. I think it's safe enough that I take it myself. Why would I tell you to take something that I wouldn't take?" Right? And so I think that it's really important when you have people who've already done it and have gone through it. And again, as has been pointed out on several occasions, there are side effects potentially to vaccines, but there are a hell of a lot more side effects to getting infected and getting sick. And you brought up the long haulers and all these other things. This is really an apples and oranges thing, right? It just doesn't compute, right? We're all in a center of higher learning. It just doesn't make sense to anybody to think about 350,000 people dead. I get a sore arm, right? Where's the balance there?
Yeah, I think that there's just a problem understanding risk versus benefit, and that's of course how decisions are made to approve drugs, whether they're under emergency use or for regular indication. And again, there's a lot of misinformation, but let's go back before the pandemic. We have a very strong anti-vaxxer movement here in California. And the rationale for their philosophy is not based in good science. A lot of it's misleading. At least it's Robert Kennedy Jr., who leads one of that. He didn't put up that fuss for the SARS-CoV-2 vaccines, but it's very difficult. And I think it does stem, quite honestly, from a lack of science literacy. And again, this goes back to the beginning of the epidemic, and when the California Department of Public Health said, no, we're not recommending using face masks because we don't have the evidence base right now to support it. And they were worried that people might feel invulnerable and, sort of reduce their distancing. But as the pandemic wore on, evidence was amassed. And I think just this idea that data, are obtained as we move along, new information becomes evident, evidence base gets built up, and your policies and recommendations change and should change based on this. And again, you can ask people now, there's some people that say, why should I wear a mask today? I mean, they're saying that. So I think that's a fundamental issue, I think, with not having good science competency and literacy. I think it goes back to elementary school. I think the idea of a propagation of an epidemic, just very simple epidemiology. We teach civics in middle school and high school. Why aren't we teaching a little bit of public health competency? I think those are going to be important. We wouldn't have as much resistance now. So,
spoken as the chair of public health, but I will also point out that people who are older, who actually saw people get really sick with measles and saw people get really sick with other illnesses that had mortality rates too, right? Or people with polio that still have problems, or died or needed to be supported on iron lungs. I don't think people who are younger, where the majority of the population is protecting their kids that they don't want vaccinated, understand that the vaccines are why they can be anti-vaxxers, right? That's why they can pull that stuff, because they're being protected by everybody else who's doing the right thing.
Yep.
Thank you. There's this question here that they like to. This question from Juan Montes Morales. What do inconclusive results stem from? When was it made a point to require that individuals do not eat or drink for 30 minutes before the test? Was it something that was implemented at the beginning of or later on? So maybe some of these points were already addressed by Richard, but maybe we'll clarify at the end here.
Okay, inconclusive results. There was the potential, if you're looking at two viral genes, N1 and N2, right at the limit of detection stochastically in a small reaction volume, you get to the point where you may detect one, you may not detect the other. And we would retest those samples, though now we've gone to a superplex where we test them both together. But if we then retested them, we still have one versus the other. We sent it back as inconclusive. Also, if the sample is no good, and it happens sometimes we don't get enough saliva, or the human control, it doesn't amplify, that's inconclusive. We decided actually to reduce the number of inconclusive tests because we put N1 and N2 together. We decided because of the time delay, it would delay the result back by a day. It was actually better just to request another sample and then get a good sample to do the test. When was it made a point not to require individuals? This came out that, well, obviously we don't want bits of Coca-Cola floating around our PCR or chunks of food. And it was made right at the beginning. If we were going to work with saliva, we didn't want people to have put anything in the mouth. We actually did do a test where members of the testing lab did various things like drank Sprite, chewed gum, and various other things. The test was remarkably robust, but There was one thing that did actually affect the test if it was done 5 minutes beforehand. I can't remember what it was. But anyway, it's really just to make sure that your saliva has not been diluted and there aren't a lot of, we don't want to test what you have for breakfast.
So there was also this question from Bruno Pedon, but Richard already alluded to that. This question is, when is these results going to be published? They are published, for example, here, let's make advertise to the community. There's a website for that that's indicated here. The dashboard is showing the results as they come and the level of infection.
Yeah, so the dashboard reports post results on a weekly basis, but the results are going out automatically. And as you see, now we're reporting about 10% of the tests are going out the same day, which I think 95% are going out next day, which is pretty impressive.
Amazing.
So I don't know if you already addressed that question or not, but there was a question about what's the follow-up if someone gets positive, if there is some arrangement with the primary care doctor. What was the follow-up if that? Maybe you or Brad could answer that part.
Well, I mean, it very quickly, it triggers all the contact tracing. You will be, as soon as the result hits point and click, the health system, someone will call you up and a whole procedure kicks into place. There's a lot of support. Yeah.
That's exactly right. And actually, of course, on the campus side, we take care of that at UC Davis. But for the community site, as Richard said, we are doing contact tracing, and actually we are authorized to do that by Yolo County through a memorandum of agreement that we have with them. But that does trigger a whole cascade of things. And the Healthy Davis Together project has actually made resources available. If people do need to isolate and they can't do that in their homes, we provide resources, both housing, food, and even wraparound social services. Communicare, we've partnered with them in the city to help provide some of those services as well. So the idea here is if we have a positive result, number one, we do the appropriate contact tracing to try to identify people that person may have come in contact with. But we also try to get the new case, out of circulation as quickly as possible so they don't infect other people. And we've had incredible success in doing this, and we've partnered with several of the businesses in Davis, including apartment owners, as well as a hotel and the city. And so this is something that I think we will be able to write with some pride that we were able to do this very successfully.
Mary Delaney, who I believe it was is the former associate dean of College of Agriculture and Environmental Science. She has a couple of questions that are already answered, and they also made a suggestion that to have a pictorial story following a sample speech to the results to be published in the Davis Enterprise so that the community would know what's going on in between that now and then. Okay, Richard. So
sure, I'm sure we can do that. Actually, we've been asked a couple of times about what happens to my spit. And
right. And another question that came a lot is that are there any other use being made of this saliva? Are they kept for other use? One line of question from one person was like my privacy thing, and another person was looking at that. Can we explore these samples in the future for some other diseases or some other things like that? So can you tell a little bit about what's the destiny of these samples?
This is obviously going back to the last question, the communications group at UC Davis, they are going to make a video of exactly what's happening in detail. So that's in production right now. So I made the point that no PHI, no public health information is attached to the tube. So once it leaves the kiosk, that's all in the HIPAA compliant database. All we get is a tube with a barcode on it. And we report the results for that barcode back to the HIPAA compliant environment. We know nothing about the individual. So there is no sort of, there's no danger to people inadvertently having their information leaked from the testing lab. The other thing is, yes, so there is a human control that we need to test that we can amplify up just a human gene that just tells us the test has worked. But There is absolutely, it's just plus or minus. It's a band on there. We know absolutely nothing about the human. So we're very, very careful. Now, the samples at the moment are kept for quality control, and also we can use them for public health, but we cannot use them for research without an IRB. Right, so if we're improving the test, or if we need positive controls, we can do that. We could sequence them to see if there's a variant for public health purposes, right? If it's completely de-identify what these samples are, we can prevent aggregate data. But as you saw in my presentation, there was absolutely no way you'd ever know that one component of a bar graph was a particular individual. So yeah, we're very, very rigorous about privacy concerns. We know nothing about the individual.
Very good. So I'm going to go back to Health Davis Together in a moment. But I would like to ask Dr. Cohen if you would like to share some of this question that is in the chat here that he's looking at, and he's responding real time. He's a very busy man who is doing multitasking, answer all these questions. Dr. Cohen, is there any particular question there that you'd like to share with us, with the rest of the audience?
No, I think many of these have been answered. I think some of them are more related to family members and things like that. One thing I think may be worth mentioning, so somebody asked about when vaccines will be available for children. So the clinical trials are at very least being planned at this point to start doing vaccine trials in kids. So it'll take a while, but it'll be there.
So I'll let you go, but before that, I would like you to once more clarify for the public in general what they can do for this vaccine. They have to wait for the health provider to give them some information, and the offices like the Yolo County in our case to provide some information. That's all they can do right now. There's no rush to go to the primary care doctor because those are not the ones that are going to be administer the vaccine, correct?
Right. So for example, at UC Davis Health, the way that it's working is we've sent out emails to all of our patients, all the people who have their primary care physician or other provider at UC Davis. They've all received my chart messages saying, we'll roll out the vaccine, and as soon as your group is ready to go, you will receive notification. For the people that we did start vaccinating yesterday, everybody who is 75 and older who we have vaccine for right now have all been notified. So we are trying to move that along. Now, when mass vaccination sites pick up in Yolo County or in Sacramento County, they're talking about opening up Cal Expo. That will be for different patient populations, probably people who aren't covered by one of the big four health systems within the city of Sacramento or within the area, right? So people either get their care from Kaiser or from us or from Sutter or from Dignity Health in this general region. But there are some people who don't get their care from any of those groups. And that's where those Cal Expo sites are going to be, sort of like Cal Expo opened up testing when the other systems were testing all their own people. And so it's another safety net. And, you know, it changes every day now as to who's now in tier 1 and how much vaccine is going to be released and everything else. So it's very hard to keep up with it. And I'm not high enough in the food chain here to actually know when we're getting more vaccine and how much more is coming. We work as an entity through the UC system. We're what's called a multi-county entity. And I think the vaccine is being distributed through the UC system. at least it has been for the Moderna vaccine, and how that ends up being spread out to patients. This is all something that's above my pay grade.
Yeah, and it is very complicated. I'll just add that across the country, the federal government negotiated directly with CVS and Walgreens to do some of the vaccination of the elderly congregate living population. And that bypassed the state health departments completely. So the vaccine distribution, which again, ultimately is controlled at the federal level, then it gets down to the state level. It is complicated right now. And I can assure you that in 8 days, that we'll have another set of vaccine distribution rules that will be in place with the new administration. So it is interesting. I think the CDC and the national academies did put together the recommended phases that we have in front of us. But in terms of how quickly we're going to get through those and rearranging things and reclassifying some groups, that's very dynamic, as the provost pointed out. We're trying to get higher education in there in the same tier as K through 12 schools. So it's evolving quickly. And again, you heard a lot about county health departments not getting a lot of vaccine. Well, turns out that most of the vaccine in Sacramento County went to UC Davis Health, it went to Sutter, it went to Kaiser, it went to the health delivery systems as the primary place. And the county ended up getting relatively few doses. And that was to cover their healthcare workers that work in some of the clinics and federally qualified clinics that wouldn't normally have access. And we are actually trying to, as Dr. Cohen said, we're trying to help out with that. But the vaccine distribution thing, it's not a simple linear thing, and it's going to be changing, I think, by the day. It certainly has over the last couple of days.
The one last thing I'll mention is that this country, and this isn't politics, Dr. Leal, this country hasn't invested in public health departments. And so when health departments have to ramp up to address disasters, they don't have people, right? If Sac County wanted to be giving out vaccines to everybody, who's doing it, right? They're asking people to come back, retired nurses to come back and volunteer to give vaccines, right? This is the health infrastructure in this country is not really built for this kind of medical disaster.
Well, it was, and I will say over the last 20 years, the public health infrastructure has been supported less and less. Again, you go back 20, 30 years ago, and I have to say, it's gone down throughout Democratic and Republican administrations. And we're in a very tough pickle right now. We don't have adequate public health infrastructure. We don't have the adequate workforce. It is something that we had a discussion with Celine Gowunder, who's Biden's COVID-19 person last week. She actually brought up this issue about the public health workforce, and we need to be looking at more creative solutions to get that up. But this has been going on for 20 years plus, and I think this pandemic hopefully will have a reset for us.
Dr. Cohen, there are many questions for you, but I know that you have something else. And you also,
I've been here since 5:30 this morning. That's what I have to just go home.
Yeah. Right. Yes, and also these people that have more questions. Dean Lymore mentioned to me that you are going to be in some sort of program this week, also some sort of chatting or something that's going to happen this week.
I think I'm supposed to be on Facebook Live tomorrow at noon.
Tomorrow at noon. So as soon
for
as
half hour.
So I encourage everyone here who has
half hour, and then I'm doing something with Sac County next week. So, I'm not very attractive to look at. I'm not sure why anyone wants to see me, and my speaking style is very halting when I haven't been asleep for too long.
It's the information that you carry, the information. And may I share something interesting? Some people that attended this symposium before, when I announced that you are going to be added to the program, they asked me, did he have time already to clean up his desk? And, and the answer is no, right?
The answer is clearly no. It's just worse now. And this is how it goes. I used to have a sense of humor too. I've lost that entirely. So thank you for inviting me,
Thank
and
you
I
for being here.
apologize for running off on you. Although
No.
you've been going for over 2 hours.
Yes.
So you have and there's still 234 people here. So
And there are a lot more on YouTube here. There's a lot more people
it's
also.
amazing. You've done an incredible job. This is always an amazingly well-designed symposium, and I'm glad to be a part of it. And I will be going to sleep shortly.
As always, thank you very much. Thank you very
got
much.
it.
And
See
now,
ya.
now let me move. Let me ask one thing to Brad. That there is a lot of question here about the Health Davis Together and whether this project could be expanded to the communities, to the other communities. And it looks like at this moment there's some difficulties there. But one thing that might be mentioned is that the Health Davis Together, the testing program, is also helping the retirement communities in the county.
The whole county, yes, that's right.
The whole county.
That's correct.
Of the city of Davis. All right.
Yeah, I think that was something, and again, Dr. Amy Sisson, who is the Yolo County Health Officer, and is actually one of my volunteer faculty in our department, one of my teaching partners. We did reach out. We have a very close relationship, and it was pretty clear that congregate living facilities and the staff that work at them, it's not just the residents, are, that we've seen the highest mortality across the country, and even here in Yolo County from the nursing homes. And so we actually worked out an arrangement now. So we are doing all of that. And we'd like to expand out. The scope of this project is pretty big. Just doing all of Davis was a very, very big task. And this is one where the incremental costs and ability to bring in another population, which are the nursing home population, where there's probably going to be a very high yield. But again, it's a reason we are a little bit resource constrained, and that's why we couldn't do the entire county from day one. We'd like to. And I live in Sacramento. I'm actually Zooming from my home here in Sacramento. I do work in Davis, but if you think about diversity and you think about some of the populations, we have other challenges here. Of course, you do in other parts of Yolo County. Davis is a wonderful community, and I will say the likelihood of success of our project is really a result of the people who live in Davis, a very educated population, very cooperative, and as I said earlier, an incredible partnership with the city of Davis, with the mayor, with the council members, with the city manager and his staff have just been incredible. And I can just tell you, this weekend, I was on equivalent of about 5 hours of calls starting Friday night through Sunday night with our Healthy Davis Together, part of our team, and also the city leaders. So they've been very dedicated to this, and I don't think it'd be possible otherwise. It's hard to generalize that directly to all the other communities in Yolo County, let alone to try to go to urban counties like Sacramento. But we hope we'll learn some things, even if it's not the entire project, but certainly parts of testing, maybe even our vaccination approach will be generalizable, we think, and will help other communities.
So the ultimate goal is to have a Davis COVID-free, which is a very difficult target to reach, but it's good to have a goal that's very high so that you can fight for that, right?
Yeah, so, and us epidemiologists, we always say all pandemics end. They may not end well, but they all end. They're not forever. An epidemic is something that is an unusual occurrence, and epidemics don't, at least infectious disease epidemics don't go on forever. I should say acute infectious diseases, because HIV is still there. But I will say that we do expect it to end. We hope that we can eradicate the virus. It may not be possible because it's an RNA virus that does mutate more. Hopefully it'll be better handled than influenza, which is very tough, where we have to guess every single year about what's coming for the next year to prepare the vaccines. I hope that we won't have that same challenge here. But the idea here is to reduce the transmission down to levels where you can't sustain it, and to increase the immune protection, the herd immunity of the population, to snuff out the virus. That's what we're trying to do. And I hope that we're just talking about one virus. We are talking about one virus now with variants, but now we've got the clock ticking in terms of, this penetration of a much more infectious version of the virus. And that the vaccines really need to be rolled out. That's how we see getting to the end of this.
So you talk about influenza vaccine, and the one reason that we still lose a lot of people to the flu is because the vaccine is only 50% effective, and they also only maybe 50% of the population gets that vaccine.
Yeah, it's the second factor that is probably a bigger driver. It is the case that if you're vaccinated in a year when we don't match the strains up particularly well with the vaccines, you still get protection against some of the much more severe complications and mortality. You do get protection, not as much, but most of the influenza epidemic numbers are driven by people who are not vaccinated, who do end up getting seriously ill. And I will tell you, it's again anecdotal, but my closest friend's daughter, 34-year-old, had 3 young daughters. She contracted influenza 4 years ago. She died in 4 days. She developed pneumonia. No known pre-existing conditions. Influenza kills a lot of people every year. Forget about the pandemic from 101 years ago. It is a killer, major killer, and it's a serious thing. And again, if we had higher influenza vaccination rates, we would be able to reduce that mortality. But it's a challenge. Like I said, it's a challenge because of the fact that that virus mutates, and there's a lot more cross-species transmission. And that's the other thing. When you go between species, there's more opportunities for these mutations that may be favorable to the virus to occur. And so influenza, we certainly know that if we talk of swine flu, we've got the bird flu, and those are examples where it's very common to have cross-species. So that contributes to more mutation, which makes it difficult to come up with vaccines that are, super effective all the time. I hope this will be different with this coronavirus.
So the testing part of the program is very successful, but could we use more people of the city of Davis to be tested as part of the program?
Yes, absolutely. And we are stepping up our mass communications campaigning in the next week or two. We really want to get people out and get them tested. So we have the capacity to do that. It will make a big difference. Again, it may be 1 in 25 or 1 in 20 people in Davis are walking around asymptomatic and capable of spreading the virus. So I think that there's a clear need to get everybody tested and to do this on a regular basis. And we're not getting there yet. Again, we may have about 50% of the people that have come in and gotten tested at least once, but we want to get everybody in there tested every week as much as possible. And so you'll see a lot more advertisements going out and some targeting for messaging to try to improve the participation rates for our testing.
So the component of the test is very successful. What's the other component that you wanted to improve as well? Is behavioral change?
Yes, and so the project, and I'm glad that's something I did. I was able to point out on the MSNBC interview with Stephanie Rule. I wanted to make a point. The testing is incredibly important. I'm an epidemiologist. The triad of what we do for epidemiology is testing, contact tracing, and quarantine isolation. Those 3 things, the triad, are really our major tool in epidemiology. But when we were putting this project concept together back in the early summer, it was very clear that we needed to also think about the health behavior change, because what has driven the epidemic hasn't just been the lack of testing, it's been behavior. And it's been not being adherent to what we know are safe practices, the public health, non-pharmacologic interventions. So Healthy Davis Together focuses equally on the health behavior change, mass communications, health education, and also measuring the impact in the community for that. So we are doing survey work to look and see how health behaviors has changed over the course of the pandemic. And that's really, really important. So we do want to have people to remain adherent and to the best recommendations for distancing, socially distancing, wearing your mask, good hand hygiene. Those are all very important. And as you heard tonight, the fact that we have people vaccinated, we still can't let our guard down. That's going to continue until we get to having a population herd immunity that's appropriate. So we don't want to let up on any of the behavior issues that we need to deal with.
So we work at the university. To come to campus, we have to be tested every 7 days. But there are people out there in the city that they say, I don't feel sick or anything, and why should I get tested? What, as an epidemiologist, what do you tell these people?
Yeah, it's really one of those issues that first and foremost, it's your kind of responsibility to everybody else. Because if you do get infected, and you're asymptomatic, you can spread it to other people and not know it. And so there's a little bit of social responsibility that we try to impart there. There's also the fact that you may be asymptomatic today. You may be what we call pre-symptomatic. Maybe you're 2 or 3 days away from having your symptoms. And so again, we just don't want people to get in that situation. It is personal risk. So trying to adhere to public health practices to cut down transmission and infection helps everybody. But in particular, if you're feeling fine, you may be feeling fine where you don't have virus in you, or you may be feeling fine when you are totally viremic and able to emit millions of particles of virus in a breath. So, and you don't know that, and you don't, unless you're tested, you have no idea. So that's the issue there. And I think just in the CDC's MMWR publication in the last week or two, there was a published article that really said that about half of the transmission occurs among people that are asymptomatic or pre-symptomatic. So that's tough. And you've heard Dr. Fauci say that's the really tough part about this particular infection. Influenza is different. Most of the viremia and ability to infect others really occur at the time that you develop symptoms or shortly thereafter. This virus, you are asymptomatic or pre-symptomatic. You've got a good number of days where you can be shedding the virus. So that's the message to get across to people. Please help yourself, help others by getting tested, knowing that you're not going to infect others, and adhere to the public health practices.
Thank you very much for what you are doing for the community. Thank you very much for being here until this late. If there is anything else that you'd like to tell about the Health Days program, the Health Days Together, please tell now. I appreciate your time to be here until this late.
Thank you. The one thing I want to say is that the Healthy Davis Together is a team effort. We have an incredible number of people, both on our executive committee membership, our advisory committee, our internal advisory committee, which is made up of a combination of university and folks from the community and from the county. And in addition, we have an external advisory committee as well. So there's a lot of people that have put a lot of person power into making the project work and be successful. And I have the honor of being the director, but it is absolutely a team effort across the university and with the support of the chancellor also. So we're really privileged to be able to do this. As an epidemiologist, I never thought that I would actually get to live through a pandemic. I wouldn't say that was an opportunity. And I will just share with you, for those of you who want to know, when I was going through my epidemiology training, starting my master's degree in 1979 at UCLA, there were 2 tracks. There was a track for chronic disease epidemiology that deals with heart disease, cancer, and everything else, and a track for infectious disease epidemiology. Only about 5% of our students, just a handful, actually did infectious disease epidemiology. It wasn't really popular back then. In my doctoral program at UCLA, just by happenstance, I took a 1-year infectious disease epidemiology elective, which I didn't need to have. And thank heaven I did, because even though I'm a cancer epidemiologist, I got retooled. And so I will say that a lot of us have kind of come out of the woodwork, but it's a pleasure to be able to try to make a difference. And so many people are doing that across the board. So thank you for inviting me. And it was a great symposium. I really enjoyed listening to everybody tonight.
Thank you very much and good night. Thank you all for participating. In special, I would like to thank you all those that participated and in terms of asking questions in the audience, and also the students and the others that provide a nice format for this symposium. I would like to send a special thanks to Kathy Garvey for the communication and Steve Dana, who is always there to help. I have some ideas about some concept, about some logo, and immediately Steve Dana provides that information that was possible. So this is a kind of event of the university, that the only committee at the university that gets unanimous consensus on everything, because I was the only member of that committee. So every decision was unanimous. But besides the university, I have also people outside, like IT personnel, Heman Patel in our department, and like I mentioned, Kathy Garvey and Steve Dana. So they're the people that are supporting and they make this thing possible. Let's keep working together to fight this pandemic. It's a turbulent time, but the blue skies are ahead. Mask up and keep practice physical distance. Stay safe, everyone. Good night.